Professor Daisuke Sano from Tohoku University, Japan,
Professor Dr. Insinyur Hefni Effendi, Master of Philosophy from IPB University Indonesia,
and also our invited speakers, Professor Dr. Gunanti Mahasri MS from Universitas Airlangga, Indonesia,
Associate Professor Dr. Yuni Kilawati from Universitas Brawijaya Indonesia,
and the respectable all invited guests and all participants.
First of all, let us praise to Allah Subhanahu wa Ta'ala who has given us mercy and blessings so we are able to attend this very special event.
Secondly, all prayers are presented to our Prophet Muhammad, the greatest leader who has guided us from the darkness to brightness.
Ladies and gentlemen, all the guests and the audience,
We would like to say,
welcome to the second Aquatic Science International Conference 2023 under the theme
Aquatic Science Roles in Supporting the Sustainable Aquatic Development.
Thank you very much for attending our invitation. I am Naila Novalia
And Benaya Tadius Garang
as the Master of Ceremony, and we would like to read today's agenda as follows.
The first agenda is opening.
The second agenda is sending the national anthem Indonesia Raya.
The third agenda is report by Chairman of the second ASIC 2023.
The fourth agenda is welcoming speech from Fisdin.
The fifth agenda is plenary session of keynote speakers and invited speakers.
And the last agenda is closing for day 1.
Before we delve even further, let us open our agenda today by praying according to our belief and religion. Please, time is yours. Thank you very much. May the agenda run smoothly and successful.
Dear ladies and gentlemen, now we are going to the second agenda, deliver our national anthem, Indonesia Raya.
By Chairman of the Second Aquatic Science International Conference 2023, to Mr. Abdul Aziz Amin, the time is yours.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen, honorable college. Welcome to the second Aquatic Science International Conference, ASIC 2023. May peace be upon you. Distinguished guest speaker, participant, ladies and gentlemen, a perfect morning to you all. Welcome to the conference, the second Aquatic Science International Conference, ASIC 2023, which will be held from today and tomorrow as the online conference from September 28 to September 29, 2023. It give me immense warmth and great pleasure to grace to all your presence in the interest of entire committee. It gave me tremendous contentment to present the welcome speech amongst the most esteemed personality who have won appreciation in their respective field. Before we begin this international conference, I would like to express my heartfelt gratitude to all of you who sincerely commit to this event to make it a success. This event would have been impossible without the support of everyone present here. This conference is organized by Faculty Fisheries and Marine Sciences, Brawijaya University, Indonesia. Ladies and gentlemen, on this particular occasion, I also would like to thank all the distinguished keynote speaker and invited speaker who come from 5 countries. The first one is Dr. Hilary Rigney from Bologna, Italia. And then second one is Prof. Gayatri Devi from Afinisingham University, India. The third one, the Professor Daisuke Sano from Tohoku University, Japan. Associate Professor Dr. Rafida Binti Othman from University Malaysia Sabah, Malaysia. Professor Dr. Hefni Effendi from IPB University, Indonesia. Professor Dr. Gunanti Mahasri from Universitas Airlangga, Indonesia. Associate Professor Dr. Andi Kurniawan from Brawijaya University, Indonesia. And the last one, Associate Professor Dr. Yuni Kilawati from Brawijaya University, Indonesia. Over 65 presenters from student, lecturer, and the researcher on aquatic science registered in this conference. Hopefully, ASIC 2023 will enhance our insight and contribute to addressing global life of aquatic resources challenges. Therefore, we proudly present this annual conference under the theme, the Aquatic Science Role in Supporting the Sustainable Aquatic Development. The second ASIC is successfully because the support of many parties, including Rector of Universitas Brawijaya, Professor Widodo, SSE, MSc, PhD, and the Dean of Faculty Fisheries and Marine Sciences, Professor Dr. Insinyur Maftu, and all dedicated committee who have been doing fantastic job to make this event run smoothly. We believe that all presenters and participants will have scientific knowledge and exchange in the next few days after joining this conference, and we are looking forward to learning about the outcome. My name is Abdul Aziz Amin. I represent from the Second Aquatic Science International Conference Committee. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much to Mr. Abdul Aziz Amin for the report and warm welcome.
Excellencies, participants, ladies and gentlemen, now we are going to listen the opening speech delivered by Vice Dean of Student Affairs, Faculty of Fisheries and Marine Science, Dr. Eng Abu Bakar Sambah, SPIMT. Time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Very good morning, ladies and gentlemen. The Honorable keynote and invited speaker of the Second Aquatic Science International Conference 2023. The Honorable speakers, Dr. Hilary Rigney from University of Bologna, Italy, Prof. Gayatri Devi from Afinisingham University, India, Prof. Daisuke Sano from Tohoku University, Japan, Associate Professor Rafidah Binti Othman, Borneo Marine Research Institute, University Malaysia Sabah, Malaysia, Professor Dr. Insinyur Hefni Effendi from IPB University, Indonesia, Professor Dr. Insinyur Gunanti Mahasri from University Airlangga, Indonesia, Associate Professor Andi Kurniawan from Universitas Brawijaya, Indonesia, and also Associate Professor Yuni Kilawati from Universitas Brawijaya, Indonesia. all the participants of the second Aquatic Science International Conference. First of all, let us praise to the presence of Allah Subhanahu wa Ta'ala, the most gracious and merciful God. Today, we can gather as online forum in the second Aquatic Science International Conference with the theme of aquatic science role in supporting the sustainable aquatic development. Aquatic ecosystem has increasingly been recognized as a center to sustainable water resources management. There are key aspects of aquatic ecosystem that need consideration during management. The influence of quantities of water and where the water comes from, the quality of the waters, the habitats that make up the ecosystem, and the biodiversity of the species that make use of these habitats. The management of aquatic ecosystem as part of water resources management involve specialist area of study such as environmental flow assessment, target setting, pollution control, water treatment, biodiversity management, and many other fields. Aquatic science provides the basis of new and sustainable approaches, solutions, and technologies to meet the challenge of aquatic sustainable development. The role of science in the implementation of the Sustainable Development Goals on water will be particularly important as tackling water challenges require adequate resources and new scientific approaches to deploy sustainable solutions and appropriate technologies. The contribution of science to the achievement of aquatic development has already been significant as the proposed Sustainable Development Goals water target built on the unfinished agenda on access to water and sanitation and link it to water resources management issues, such as improving water quality and water management, increasing water efficiency, promoting integrated water resources management, and reducing water-related risk. The contribution of the academic community is central to developing sustainable solutions to address water challenge through advancing aquatic science, research, and knowledge, and applying them in the preparation, establishment, and testing of the appropriate frameworks and tools that would be needed to address the challenge in the aquatic sector. Through this conference, academicians, scientists, practitioners, policymakers, students, and local community can share information, experiences, and knowledge to each other about sustainable aquatic resources. I hope this conference will be the beginning of a mutual beneficial collaboration between researchers, institutions, and stakeholders in developing sustainable aquatic resources. I'm honored to be here at this remarkable meeting, and I'm looking forward to hearing excellent and productive discussion within this conference. And also congratulations to the committee who have tried and worked hard in preparing for this international conference. Thank you very much. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much for the opening speech, Dr. Abu Bakar Samba.
Well, ladies and gentlemen, we are about to enter the fifth agenda of this morning, which is the plenary session of keynote speaker. We would like to invite the moderator for this plenary session to Dr. Umi Zakia. Please, the time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Morning, dear all. Honorable Dean and staff of Faculty of Fisheries and Marine Science, Brawijaya University. Honorable guest speakers in the second Aquatic Science International Conference. Distinguished participants, ladies and gentlemen. Let me introduce myself. I am Umi Zakia. It gives me a great pleasure to be the moderator for the plenary session in this conference. As we know that aquatic science has become the basic issues in regard with aquatic sustainable development and management due to its function and crucial role in human welfare. Undeniable, have been rapidly developed. So in today's conference, we will get an insight into those knowledges, including the technology involved, methods, implications, risks, and opportunity that valuable to us, to students, lecturer, researcher, or even the scientific community involved parties to hear. So, ladies and gentlemen, first I'd like to read a brief introduction to our first speaker, Prof. Dr. S. Gayatri Devi. Hello, Prof. How are you today?
Hello, madam. I'm fine here. How are you?
We meet again, huh?
Yeah, definitely. It's really nice to meet you again in the conference. It's really happy to be here.
Yeah.
Yeah,
Okay. Us too.
yeah, definitely.
Okay. As you acknowledges your curriculum vitae, it's very long. So let me just say it in a short
Definitely.
way.
Yeah, sure, sure.
Professor S. Gayatri Devi is from the Department of Biochemistry, Biotechnology, and Bioinformatics, Avinashilingam University, India. Specialization, novel compound isolation from medicine plant gene expressions in silico for their anti-diabetic, anti-obesity, antioxidant, and cardioprotective activities. In environmental studies, bioremediation of electroplating effluent, biodegradation of HDPE using microbes. And international visiting professor for the period 1-1 to the Department of Biology, Brawijaya University, Malang, Indonesia, from 1st October to 31st October 2019. And funded project, principal investigator, Dr. G. P. Jayanti, co-investigator, Dr. S. Gayatri Devi, post-translational changes in human lens protein among Ethacaritus diabetic subject UGC, funded 11.93 lakhs, 3 years for in 2011 till 2014. Co-investigator Dr. S. Gayatri Devi, efficacy of Nutrimate on iron status of selected rural adolescent girl in Coimbatore, H.E. Heinz Company Foundations, US, 14.57 lakhs, 30 months, in 2013 until 2015. Publications, more than 40. Book chapter 5, conference publication 23rd. Research person invited lecture 17. So I hope that will be enough, Prof. Gayatri.
Yeah,
Okay,
ma'am, yeah.
without further ado, Now let us hear the material from Dr. Gayatri, Professor Gayatri in the topic of aquatic environmental study based on biotechnology. Prof. Gayatri,
Yes.
you can deliver your topic in about 45 minutes, and then we will have a Q&A sessions for another 45 minutes.
Yes.
So now time and place is yours.
So very good morning to one and all present here. It is indeed very happy to be here, and I feel it is a proud moment to address you all in the aquatic environmental study, and for this marine sciences conference, international conference. In fact, I am very, very thankful and would like to show my gratitude to Professor Sasmito, and then present current director Dr. Vidadu, and then my sincere gratitudes are also due to Dr. Abubakar, and then Dr. Oun and Dr. Umi, and in fact Dr. Laras. Because of them only I am able to have very good rapport with Indonesia, and of course Dr. Kalaiselvi from my part from India. So I thank God Almighty to be here to present this paper, that is a topic on aquatic environmental study based on biotechnology point of view. In fact, I thank all the organizers as well as the participants of this conference. Okay. Can I have the next slide, madam?
Yeah.
Yes. So this presentation, mainly I'm going to concentrate on the wonders of aquatic environment and why aquatic environmental studies are important, and how we are going to explore the aquatic biodiversity, challenges in aquatic environmental studies, future of aquatic environmental study, and how to make a difference in the aquatic environmental studies. So about which let us go to see now. Next slide, please. So, did you all know that over 70% of the Earth's surface is covered by water? Yet, we have only explored less than 5% of it. There is a whole world beneath the waves waiting to be discovered and studied. The aquatic environmental studies allows us to explore this vast underwater world and understand the complex ecosystems that exist within it. From microscopic organisms to massive whales, every creature plays a vital role in maintaining the delicate balance of our planet's aquatic environment. Next slide. So why aquatic environmental studies are important? Aquatic environmental studies are very crucial in understanding the complex interactions between the various species and their habitats. So by studying these ecosystems, we can gain insights into the delicate balance that exists within them and the impact of human activities on their health and sustainability. So the human activities such as pollution, overfishing, and climate change have had a significant impact on aquatic environment. So these activities have led to the loss of biodiversity, altered food webs, and the degradation of critical habitats. Though through this, the aquatic solutions to address them by environmental studies, we can identify the causes of these issues and work towards developing. Next. So how to explore this aquatic biodiversity? Aquatic ecosystems are home to an incredible array of biodiversity, from tiny plankton to massive whales. In fact, more than half of all known species on Earth live in this aquatic environment. These organisms play a crucial role in maintaining the health of planet's ecosystem, including production of oxygen, regulation of the climate, and providing the food for human and other animals. Unfortunately, human activities such as pollution, overfishing, and habitat destruction are threatening this rich diversity of life. It is essential that we take action to protect these fragile ecosystems and the creatures that call them home. By implementing sustainable fishing practices, by reducing the pollution, by conserving the wetlands and other critical habitats, we can help to ensure the survival of aquatic biodiversity for generations to come. Next, madam. So what are the challenges in aquatic environmental studies? So we have to study the aquatic environment presents a unique set of challenges, and unlike terrestrial environment, water is constantly moving and a changing medium that can be difficult to monitor and measure accurately. Additionally, aquatic ecosystems are often complex and interconnect, making it challenging to isolate specific factors and understand their individual impacts. Furthermore, human activities such as pollution and climatic changes have significant impact on aquatic systems, further complicating the study of this environment. It is essential that we find innovative solutions to address these challenges and ensure the long-term health and sustainability of our planet's aquatic ecosystem. Next one. The future of aquatic environmental studies, as we move forward into the future, there is a great potential for advancement in technology and research to revolutionize our understanding of the ecosystems. From new underwater exploration tools to advanced genetic sequencing techniques, there are countless opportunities to deepen our knowledge of the complex environment. This increased understanding will not only help us better protect and preserve these precious ecosystems, but also aid in the development of sustainable practices that can benefit both the human population and the environment at large. Next. So how we can make a difference in the aquatic environmental studies? One way to make a difference in the aquatic environmental study is by supporting the organizations that work towards preserving these ecosystems. This can be done through donations or volunteering time to help with the conservation efforts. And another way is to get involved by reducing the personal impact on aquatic environment. This can be achieved by using the use of eco-friendly products, reducing water usage, and properly disposing the waste. Next. So we are going to explore the aquatic environment. Next, madam. So here we are going to see about what are aquatic environmental studies and how biotechnology is being used in the aquatic environmental study. What are the different types of research that is going on in this environmental studies, as well as the different challenges and opportunities. Next. So what is actually aquatic environmental study? Aquatic environmental study itself is a field of research that focuses on understanding and protecting the health of aquatic ecosystems, such as rivers, lakes, and oceans. So this involves studying the complex interactions between living organisms and their physical and environment, and how these interactions are affected by human activities. The importance of aquatic environmental studies cannot be overstated. Our planet's water resources under increasing pressure from pollution, overfishing, climate change, and other factors. So by studying and understanding these issues, researchers can develop the strategies to protect and restore our waterways, ensuring that they remain healthy and productive for generations to come. For example, studies have shown that reducing the nutrient pollution in waterways can help to prevent the harmful algal blooms, which can be toxic to humans and wildlife. Next. So biotechnology, how it is going to play a main role in aquatic environmental studies? Yes, of course. Biotechnology plays a crucial role in understanding and protecting our planet's aquatic environment. By means of using genetic engineering, the researchers can develop new tools to monitor the water quality and also detect the harmful pollutants. For example, our scientists have developed biosensors that use genetically modified bacteria to detect the toxins in the water. These biosensors are highly sensitive and can detect even the trace amounts of pollutants, making them a valuable tool for monitoring the water quality. In addition to monitoring the water quality, biotechnology is also being used to develop the new treatments for waterborne diseases. One promising approach is the use of bacteriophages, which are bacteriophages or the viruses that can infect and kill the bacteria. So the researchers are exploring the use of bacteriophages to treat the diseases like cholera and typhoid fever, which are the major public health concerns in many parts of the world. Next. So what are the current research in aquatic environmental studies? So our area of current research in aquatic environmental studies is the impact of microplastics on the aquatic ecosystems. Microplastics are the small plastic particles that have accumulated in the ocean and the waterways, and they can have a harmful effect on the marine life. So researchers are studying the sources of microplastics, their distribution in aquatic environment, and their potential impact on the human health. Another area of research is the use of bioremediation. So bioremediation to clean up the contaminated waterways. And the bioremediation involves the using of microorganisms to break down the pollutants in the water, turning them into harmless substances. And this method has been successful in cleaning up the oil spills, and the researchers are now exploiting its potential for cleaning up the other types of pollutants. Next, please. So we are going to see the tools for the aquatic environment. What are the different tools? Next, please. The DNA sequencing techniques, CRISPR-Cas9 gene editing, microbial ecology tools, and remote sensing techniques. These are some of the techniques which we are going to concentrate on. Next, please. First, we are going to see about DNA sequencing technique. It is an essential tool in aquatic biotechnology research, allowing the scientists to investigate the genetic makeup of organisms and understand how they interact with their environment. There are several different DNA sequencing techniques used in aquatic biotechnology research. This includes Sanger sequencing, next generation sequencing or NGS, and metagenomics. Each technique has its strengths and limitations in choosing the right method, and it depends upon the research questions being asked for. For example, NGS, that is the next generation sequencing. This allows for a high throughput sequencing of large amount of DNA, while metagenomics can be used to study the entire microbial communities. Understanding the genetic diversity of the aquatic organism is crucial for understanding how they adapt to and respond to the environmental changes. The DNA sequencing can reveal the important information about the evolutionary history of species and help to identify the genes that are involved in the important biological processes such as nutrient cycling and pollutant degradation. Next, please. Next technique which we are going to see about is CRISPR-Cas9 gene editing. This is the current technique which is being used to modify the genes, not only in aquatic organism, in almost all the field this is being used. So here in the case of aquatic environmental studies, this CRISPR-Cas9 is a powerful gene editing tool that has revolutionized in the field of biotechnology. Its ability is to precisely target specific genes has opened up new avenues for the research, including in the field of aquatic biotechnology, and to enhance their ability to survive in changing the environmental conditions. In aquatic biotechnology research, this technology allows the scientists to make precise changes to an organism's genetic code, which can have a significant implications in the field. This could have significant implications for both the conservation efforts and aquaculture industries. For example, they can use the CRISPR-Cas9 to create a fish that are more resistant to diseases or that grow faster, leading to increased food production and improved sustainability. Next. So the microbial ecology tools is one of the important tools for studying the microbial communities in the aquatic environment is metagenomics. This technique mainly involves the sequencing of DNA directly from the environmental sample, allowing the researchers to identify the microorganisms present and understand their functional roles in the ecosystem. Another useful tool is table isotope probing. So here, this can be used to identify the specific microorganisms responsible for particular functions within the ecosystem, such as nutrient cycling or degradation of the pollutants. In addition to these molecular techniques, the researchers also use microscopy to visualize the microbial communities in situ. So fluorescence in situ hybridization FISH is an efficient method, is commonly used technique which allows the researchers to visualize the specific group of microorganisms using fluorescent labeled probes. The other technique is using electron microscopy, which can also be used to observe the microbial communities at high resolution, providing an insight into their structure and organization. Next, please. The next technique is remote sensing technique. It is a technique that is becoming increasingly important in aquatic biotechnology research. These tools allow the scientists to monitor the aquatic ecosystems from a distance without disturbing the delicate balance of the environment. One such technique is satellite imagery. This can provide a detailed information about the temperature of the water, color of the water, and other important factors. Apart from this, another technique, LIDAR, that is light detection and ranging. Here we are using the laser to map the landscape and identify the changes over time. These tools are essential for understanding how aquatic ecosystems are changing and what we can do to protect. Next, please. This next. Yes, now we are going to see about the different aquatic biotechnology methodology growing field, that is bioprospecting, phytoremediation, and how environmental sustainability could be used in this case. So next, please. Bioprospecting in aquaculture. It mainly refers to the exploration and discovery of new biological resources such as plants, animals, and microorganisms for their potential applications in various industries, including aquaculture. In the context of aquaculture, bioprospecting is mainly involved in species discovery. So, what is species discovery? Identifying the new species of aquatic organism with desirable trait for aquaculture, such as high growth rate, disease resistance, or tolerance to a specific environmental conditions. Second one is natural products. So extracting and studying the bioactive compounds from aquatic organisms for their use in fish health management, nutrition, and water quality control. Third one is genetic resources. So collection and preservation of the genetic material from wild population for use in the breeding programs to improve the formed species. So hence the bioprospecting can lead to the development of new aquaculture technologies and practices that enhance the production and sustainability. Next please. So phytoremediation, in what way phytoremediation can help in aquaculture? So what is phytoremediation first? Phytoremediation is an environmental friendly and cost-effective technique that uses plants to remove, stabilize or detoxify the pollutants from water and soil. In aquaculture, phytoremediation can be applied in many ways. First one is water quality improvement. So some of the aquatic plants such as water hyacinth and duckweed, they can absorb the excess nutrients, example nitrogen and phosphorus, that are present in the pond or effluent. Apart from that, it can also absorb the metals that are coming from the effluent. So this helps to prevent the water quality degradation and reduces the risk of harmful algal blooms. The second thing is metal and contaminant removal. Certain aquatic plants can accumulate heavy metal and contaminants from the water, making them suitable for removing the pollutants from aquaculture systems, and especially in the cases of water quality where it is to be compromised. The third one is by means of habitat enhancement. Planting have native vegetation around aquaculture facilities, which can create buffer zones that improve water quality and provide habitat for beneficial insects and microorganisms. Next, please. So this aquatic biotechnology is nowadays a rapidly growing field that has the potential to revolutionize the way we approach the research and development. With advancements in technology and an increase in understanding of aquatic organisms, researchers are able to discover new ways to harness the power of these unique organisms for a variety of applications. In fact, according to the recent data, the global market for aquatic biotechnology is expected to reach $5.9 billion by 2027 with a compound annual growth rate of 10.3%. So increased demand for sustainable food sources, the need for new medical treatment and the desire to protect and preserve our planet aquatic system is needed. Next. So how this is being helpful in sustainability of the environment? So aquatic biotechnology is also a rapidly growing field that has the potential to revolutionize our approach to environmental sustainability and the protection of aquatic ecosystems. So by leveraging the unique properties of aquatic ecosystem organisms, researchers are developing new innovative solutions to some of the biggest challenges facing our planet today. One example of this is the use of microorganisms in wastewater treatment, bacteria to break down the pollutants, and they have also developed cost-effective and environmentally friendly methods for treating the wastewater. Another example is development of biodegradable plastics which are made from algae, which can help to reduce the amount of plastic waste in our waterways as well as ocean. Next. So what are the aquatic alarms, biomonitoring and bioindicators? Let us see about it now. Next. So here we are going to see about what is biomonitoring, the types of biomonitoring, their applications, challenges and limitations. Next. So biomonitoring, it is a practice of using living organisms to assess the health of an ecosystem. In aquatic environment, this often involves in studying the populations of fish, the macroinvertebrates, and the algae to determine the overall health of the waterway. So by monitoring these pollutions over time, scientists can detect the challenges and identify the potential problem before they become too severe. So one example of biomonitoring in action is the use of macroinvertebrates such as bioindicators. These small creatures such as mayflies and stoneflies are sensitive to changes in their environment and can be used to assess the water quality. For instance, if a stream has a healthy population of macroinvertebrates, it is likely that the water quality is good. However, if the population is low or dominated by pollution tolerant species, it may indicate that the water quality is poor. Next, the different types of bioinvertebrate indicators includes fish as a bioindicators, mainly in the aquatic environment. So, they have got different tolerances for environmental changes, and they are highly sensitive to changes in the water temperature, and they are more tolerant. However, using fish as a bioindicators can be challenging due to the factors such as migration patterns and variability in population. Algae can also be used as a bioindicators, particularly in the marine environment. It responds quickly to the changes in the environment and also give early warning signal of potential problems. They are influenced by factors such as nutrient availability, water flow, which can make interpretation of data more complex. Next. So what are the applications? The biomonitoring has become an essential tool in assessing the health of aquatic environment. One of the most important application is in assessing the impact of the pollution on water quality. So by analyzing the presence and abundance of the bioindicators such as macroinvertebrates, these can determine the extent of pollution and identify its sources. This information can then be used to develop the strategies for reducing and eliminating the pollution. The next important application of biomonitoring is monitoring the effect of climatic change on aquatic system. There is a temperature rise and weather pattern shift, and the composition and distribution of aquatic species can also change, which can have a significant impact on the overall health of the ecosystem. So by monitoring these changes in the abundance and distribution, the bioindicators over time, the scientists can better understand the effect of climate change on aquatic environment and therefore develop the strategies for mitigating these effects. Next, please. What are the challenges and limitations? So biomonitoring and bioindicators are powerful tools for assessing the health of the aquatic environment, but they are not without their challenges and limitations. So one major challenge is the need for specialized expertise to properly identify and interpret bioindicator data. Without this expertise, it can be difficult to accurately assess the health of an aquatic environment. Another challenge is the potential for false positive and false negatives. Bioindicators can be affected by a variety of factors, including natural variability and human activities such as pollution and habitat destruction. This can lead to inaccurate assessments of environmental health, which can have serious consequences for conservation and restoration efforts. Next, please. So we are going to see about the power of biological sensors and genetic engineering. Next. So wastewater treatment, which turns the pollution into a research, and biological monitoring is keeping our waterways clean. Aquaculture is sustainable seafood for a growing population, and biological sensors is needed for monitoring our environment in real time. Microbial fuel cells, which harness the power of microorganisms, environmental genomics and genetic engineering is the advances of our understanding of the natural world. Next, please. So how wastewater treatment can be help in turning the pollution into a resource. So wastewater treatment is a crucial process that helps to protect our environment and ensure the health and safety of our communities. By means of removing the pollutants and contaminants from wastewater, we can turn what was once seen as a problem into a valuable resource. Not only does wastewater treatment helps to prevent the pollution and protect our water resources, but it also has a potential to generate energy and create new products. So through innovative technologies and sustainable practices, we can transform the wastewater treatment into a powerful tool for environmental and social progress. Next, please. So aquaculture for the sustainable seafood for a growing population. Aquaculture is the practice of cultivating fish, crustaceans, mollusks, and other aquatic organisms in a controlled environment such as tanks or ponds or even cages. This is mainly done for the production of seafood and also to provide a sustainable source of protein for growing population. This method is more efficient and less damage to the environment. It allows the controlled breeding and feeding of aquatic organisms, reducing the pressure on wild populations and reduce the impact on marine systems. And we can also use antibiotics and pesticides in aquaculture. And this is important to address these concerns through responsible and sustainable practices. There is an increasing demand for seafood while minimizing the impact of the environment and ensuring that the aquaculture remains a viable solution for providing a sustainable source of seafood. Next, please. Biological sensors, they are the cutting edge technology which is used to monitor our environment in real time. These sensors use living organisms such as bacteria or cells to detect the changes in their surroundings and transmit the information to a computer or other device, and environmental conditions such as pollution levels or changes in the temperature can be tracked. The potential applications of biological sensors are vast. For example, they can be used to monitor the water quality in rivers and lakes, alerting us to potential contamination before it becomes a problem. And it is also being used to detect the changes in the air quality, helping us to identify the areas where pollution levels are high and to take actions and reduce emissions. Additionally, they could also be used in agriculture to monitor the soil conditions and optimize the crop yields. Next, please. Microbial fuel cells or other methodology to harness the power of microorganisms to generate electricity. These devices use bacteria and other organisms to break down the organic matter to produce an electric current. They are also used to provide a sustainable energy source for a variety of industries and used to power the wastewater treatment plants and reducing their reliance on fossil fuels and lowering their carbon footprint. This method is also used to power from remote sensors and other low power devices, eliminating the need for batteries or other external power sources. Next, please. Next, please. Yes, the environmental genetics, genomics. This methodology uses DNA sequencing analysis to study the genetic diversity of organisms and their natural environment. The genome of bacteria, fungi, plants, and animals in their natural habitats. Researchers can gain insight into how these organisms interact with each other and surroundings. This genetic engineering methodology also involves the modifying DNA of organisms to achieve specific goals, such as improving the crop yield or developing new medical treatments. This has potential to revolutionize mainly in industries, including agriculture, medicine, and energy production. Next, please. Next. So we are going to see something about synthetic hormones to transgenic fish. Next, please. So we are going to see how synthetic hormones are induced in breeding, then about transgenic fish, gene banking, uniparental and polyploidy population and management, and health management. We are coming to the end of the session presentation. Yes, next, please. So how synthetic hormones play a vital role in induced breeding is the process of artificially stimulating the fish to breed. So increase production. This technique is mainly used to increase the production and improve the quality of fish stock by means of controlling the timing and frequency of breeding. Farmers can optimize their yield and reduce cost. The main benefit of using synthetic hormones in induced breeding is that allows the farmers to produce more fish in less time, and the hormones can also stimulate the fish to spawn at a specific time, which means that all the eggs are fertilized at once. Additionally, using synthetic hormones can also improve the quality of the fish produced. For example, farmers can use hormones to ensure that only best quality fish are bred in, resulting in higher value of offspring. Next, please. The transgenic fish, it is also known as genetically modified fish, where foreign DNA is introduced into their cells, revolutionize the aquatic culture by means of improving the growth rate, disease resistance, and nutritional value. Aqua Advantage salmon. This has been genetically modified to grow twice as fast as conventional salmon. So increased food production and reduced pressure on wild fish population. This also raises the concerns about environmental impact and safety of consuming genetically modified organisms. Next. Gene banking. What is it? It is a critical tool in preserving the genetic diversity of the aquatic species for a long-term survival and sustainability. And also it is being used for storing the genetic material from wide range of species so that we can ensure the valuable traits that are not lost due to the environment or human-induced factors such as overfishing, habitat destruction, or climate change. In addition to preserving the genetic diversity, the gene banking also has got practical applications which allows us to selectively breed fish with a desirable traits such as disease resistance, growth rate, and then feed conversion efficiency. This can lead to more sustainable and profitable aquaculture practices while reducing the need for harmful chemicals and antibiotics. Next. Next, uniparental and polyploidy population management. These are all the innovative methods of population management in aquaculture through the use of uniparental and polyploidy techniques. The uniparental reproduction involves using only one parent to create the offspring, while polyploidy involves increasing the number of chromosomes in organism cells, increased growth rate, disease resistance, and improved survival rates. Then the example is salmon farming, which is an example for uniparental reproduction, which has been used to produce all female populations, which grow faster and more disease resistant than mixed sex populations. Similarly, polyploid oysters have been shown to have higher growth rate and better survival rates than their diploid counterpart. So these techniques have the potential to revolutionize the aquatic industry, improving the efficiency and sustainability. Next, please. Coming to the health management, in aquaculture, maintaining the health of a fish is crucial for the success of the industry. So biotechnology has the potential to revolutionize the health management by providing the innovation solutions to disease prevention and treatment. And genetic engineering is help to develop the disease-resistant fish that lead to susceptible infection. This not only reduces the need for antibiotic, but also improves the overall health and welfare of the fish. And use of probiotics that are beneficial for the bacteria that can be introduced in the fish environment to promote healthy gut microbiota and improves digestion. And this can also lead to a better nutrient utilization, growth, and overall performance of the fish. Additionally, probiotic can also reduce the risk of disease outbreaks by outcompeting the harmful bacteria resources. Yes. We have come to the end of the session. So thank you one and all present. Terima kasih.
Terima kasih kembali. Thank you very much, Professor Gayatri, for giving such an informative and comprehensive presentations. Wow. I might have a difficulties to summarize. I hopefully that everybody has a joyful hearing in following this lecture just now. But maybe I can state some underline, which is I thought very important for us to understand why this aquatic sciences is very important as we acknowledge that the wonder world, aquatic environment is a wonder world, since among 70% of the Earth's surface, only 5% has been studied, and it is still going more. So that is why very important, since it's crucial, valuable, and impact on these environments will definitely impact on human welfare as well. So that's why we need, or we are challenges to explore what is inside these aquatic environments using such an advanced technology that has been done until now, and which is I cannot mention one by one, of course. Yeah, so you can hear that. So hopefully, with everything that has been Professor stated in this lecture, you would get benefit, and also you would have more wide knowledge about what is and what should we do with aquatic environments. Thank you again, Prof. Gayatri. Ladies and gentlemen, now we come to a session of discussion, question and answer, and we are glad to receive questions from the participants. Either you can raise your hands or otherwise state it in the chat so I can read it for you, or otherwise you can deliver it yourself. Now I open for the QA sessions. Please, if you have a questions or discussion or comment, we'll be glad to receive it. And we still have plenty of time to have these discussions, about 45 minutes. So please use this valuable time for you if you have any questions. Any questions?
I think we have got some questions in chat box, Dr. Omi.
Oh, okay. Yes, Prof. Gayatri. It's from Renata Ekawira. Actually, I will be glad if you introduce yourself. From where are you, students, or are you researchers from somewhere? But it's okay, maybe later she can write it down. The question: great presentation, Prof. Gayatri. We know artificial technology like GMO, artificial enzyme, hormone, etc. can contribute to destroy environmental and their organisms. How to prevent this effect to your aquatic ecosystem, to our aquatic ecosystem? So what is your opinion, Prof. Gayatri?
Yeah, of course, we know very well that always artificial technology, we cannot totally rely on that. It will be sometimes become adverse, give adverse effect also. But nowadays, this artificial enzymes and hormones has got a very good impact on the health management of an individual for humans, etc., etc. And then though we can say that some of the genetically modified organisms are harmful, and that is a great debate going on between the scientists for having whether to have this GMO or not. So totally one big debate is going on. Still for the welfare of the planet, we have to go in for these artificial technologies. No, one way it is not very good. Otherwise, what we have to do, we have to be aware very much to not to pollute the environment, not to pollute the ocean. And if you are polluting the ocean, naturally it is affecting the fish and everything, we know very well. So one way by means of reducing the pollutants, one way by means of conserving the water resources. So by that way we can do, and as well as somehow we should have an awareness not to destroy the aquatic ecosystem. So here, as per the question, we know very well that artificial technology is not that way good for the environment. But nowadays, the scientists are having a concern in preparing the GMO so that they are seeing to that it is not very much affecting. So we can go for this artificial technologies. Yes, next question.
Okay, thank you for your answer. I hope you can get the answer, Renata. So basically, not every technology is good for the environment, isn't it that, Professor?
Yes.
So we have to really choose the one that good for the environment, I suppose. Yeah. Okay. There's another questions. Oh, yeah. Again, I do hope for the participant who raise the questions, please introduce yourself. Are you from Indonesia or from Malaysia or from any place, other places, so that we can know you. Okay, this second questions from Salama.
Yeah.
Maybe this is from Malaysia. Good morning, Prof. Gayatri. Facing all the environmental problem and challenges, what kind of simple knowledge should we give to younger generations to prevent more damage? Thank you and
Yeah.
have a nice day. Okay.
Yeah, this is of course a wonderful question because the younger generations from the kids, if we started giving awareness, naturally they will have more mm, what to say knowledge about not to destroy how not to destroy the environment like that. So say for example in India we have really started now itself for around say sixth grade to students, from fifth to sixth grade students itself. We are keeping on conducting competitions like poster making or even presentations and science competitions, all those model making, everything we are doing so that they from the younger age itself they will have a conscious and they are saying we should not put this paper here, we should not throw plastics into the ocean. So they are having more awareness and they have stopped doing that. So in this way, if we give awareness from the younger generation itself, we can prevent more damage by means of conducting awareness camps or by means of conducting a rally or by means of conducting, because scientific talk and all they are not in a age to listen as well as to understand, whereas by means of simple ways like being keeping the posters or what to say just slogans like that, we can create awareness to the kids so that they will have a mind set up so that they won't destroy the ocean or any waterways as well as the in earth nature. Yes, next, madam.
Okay.
Yes.
Yes. Thank you so very much.
Yeah.
So, Salama, that's the answer from Prof. Gayatri. Yeah, I guess I'm completely agree that we should increase awareness since the beginning so that they will understand from even younger age the important things of our environments. Thank you, Prof. Gayatri. And I guess we, before we continue to uh, the question in the chat. Oh, okay. I think Mr. Aditya Yanuar want to deliver the question himself.
Yes, yes.
Okay, time is yours, Mr. Adi.
Okay, thank you for the opportunity. If my voice is okay?
Yeah, we can hear
Okay,
it clearly.
I'm going to ask about bioremediation. So that theoretically, bioremediation is a very, very promising method to overcome pollution. But in fact, bioremediation is relatively not effective to degrade pollution because it needs longer time. And usually, also it's not easy to control biological agent during bioremediation process. according to you, how is the future of bioremediation?
Yeah, it's a very good question, Mr. Aditya. I appreciate your question. Actually, we are doing in our lab itself, we are conducting some of the research which include phytoremediation. So, as I have mentioned in the talk, water hyacinth and other water plants like Salvinia natans and all, they are able to take up the contaminants as well as metals from the effluents, because industrial effluents, everything they are just throwing into the waterways only. So what we have done is we have just grown a set of plants in that particular pond and other waterways, and we just took the plant material and then we have analyzed it in the laboratory, and surprisingly we got a results that the metal we found get stored in the tissues of the plant. So we cannot very well say that this bioremediation is not effective to degrade the pollutant, because it is doing its own way. Similarly, microorganisms also degrade the contaminants. So we cannot just like that say that it is not doing. It is doing. But the thing is, as you have mentioned, it is a time-consuming process. So what we have to do, better instead of going for treatment, bioremediation and all, before throwing the garbage or effluent into the water, we have to treat it. The wastewater treatment setup has to be there in every industry. So we should see to that it has to be there and it is effectively working there. So all those things we have to see. And before disposing the waste and other things into the pond, humans should have a conscience that they should not do it. And just like that, having some Coca-Cola or something like that and throwing the pet bottle into the river as well as other waterways, that and all is not at all advisable. So we have to give an awareness programs and something like that, and to avoid the public to do this. So that way we can do this. But bioremediation in one or the other way, it is effective only. We cannot very well say that it is not at all effective, because many research papers which I have gone through is clearly saying that bioremediation is an effective process. Hope you agree with me now.
Okay.
Okay, how is it, Mr.
Uh,
Adi? What do you
Very,
think?
very interesting insight. Yeah. Thank you, Professor, for your response.
Thank you. Thank you.
Okay, thank you, Mr. Adi, for the questions. Yeah, and then now the next question is from Mr. Heather Jamaluddin. Would you like to deliver your questions yourself?
Okay, thank you, Dr. Umi Zakia. Okay, if you're talking about the environment, especially for the seawater or freshwater, there are so many kinds of problems. One of them that become the attention of the world around the world is the problem about microplastic or nanoplastic. Yeah, that damage some aquatic organism like a fish and also other organism. I'm asking about the development of this research in India to solve or to overcome these problems, because some articles that I ever read, it's only determine the number of microplastic in like a viscera or liver, and even in the edible portion of fish. How far the development of microplastic research in India? It's only determining the number of microplastic, or any any problem solving that already developed well in India. Thank you.
Okay. So I think your question is about how research in India goes on microplastics. Am I right?
Yeah, yeah.
In aquatic culture. Am I right?
Yes, yes.
So around, of course, totally around some researches are going on in the how to say that microplastic they are doing. There is abundance of microplastics in aquaculture. The products are generally higher than that of the wild aquaculture products. And cultured mussels, they have done some work on aquatic mussels around 1.4 to 1.7 times higher than the wild mussels. Like that, many researches are going on, but how far we can have a better, that is, they've come to the some aspect like bioremediation or phytoremediation. We can very well say this much percentage of work has gone, but I'm not very sure about how far work has gone in India related with the microplastics, but they are doing, I know. But as per your question, exactly the percentage, how much they have gone, I'm not sure. But around, they have reported microplastic particles ranges around 0 to 1,000 particles are there. Some reports are there, but exact answer, I'm not sure.
Yes. Still near. So that's the answer from Prof. Gayatri. Mr. Heather, do you get the answer?
Yeah.
Or do you agree or still have a comment?
Yeah, I have still a comment. Thank you, Mrs. Umi Zakia. Since microplastic is the one of the main problems around the world, but until now, there is still limitation in microplastic research, such as how to solve, how to overcome this serious problems in the world, because you know, it is not easy, right, to decrease the number. Even we applied some policies or some regulations to stop drawing the plastic into the seawater, something like that. But thank you, Mrs. Gayatri, for the insight.
Thank you.
Yeah. I guess, yeah, there is still a lot of questions. Okay. Okay. We can continue to the questions. Thank you, Mr. Heather, for the questions and Prof. Gayatri for the answers. Now we come to the next question from Nachwa Ashifa. Okay. Thank you, Prof. Gayatri. I'm a student of UP, University Brawijaya. I would like to ask questions. Are there any negative impact for us if we are eating the transgenic fish? What an interesting questions. Okay, please, Prof. Gayatri.
Yes, so Najwa's question is any negative impact for us if we are eating transgenic fish? Yes, Najwa, actually, according to FDA, there is no food safety concern or human health concern. And no nutritional differences also between the genetically engineered salmon and its conventional counterpart. So I think so it doesn't mean that the GM technologies are void of concerns. Can proceed.
So
yes,
there are still uncertain about the effect,
yes,
Prof.
yes,
Gayatri, isn't
yes.
it? Yeah, maybe there will be needed longer research for that to come to a result.
Yes.
Thank you, Prof. Gayatri. And probably to the limitation of the time, we come to the last questions
Yes.
from the chat. It's from Saifullah. Thank you for your wonderful presentations. My name is Saiful from Indonesia. Okay, I have question about polluted environments. As far as I know, that river in the big city of Indonesia, they have been polluted and will be found so many plecostomus, or we call it in Indonesia ikan sapu-sapu, or there's a type of species of fish from in a river, Prof. Gayatri, and hardly found any kind species in the river. Let's say if we can stop the source of pollutant, what is the first move or key to conserve river and bring back many diverse species in the river? So probably, yeah, how to bring back the better environment to the river.
Yeah, of course.
Prof. Gayatri.
Yeah, of course. This is a normal answer, which I'm repeatedly saying that the human should have a concern about the nature and they should have an awareness. So we should see to that. Better not to use so many vehicles. That's what. But of course, it will be very difficult for us to handle nowadays. But we can prefer going using a public transports. And we should not litter because air pollution there it increases, and we should not litter near the water bodies. We should not throw garbage and other things near the water bodies. And then so there are different ways to prevent the water pollution, like sewage. As I said earlier, the sewage should not be directly discharged into the water. Instead, we can recycle it and then it can be discharged. Before discharging, the quality control should be there, and the quality control persons should check for the microorganisms as well as other unwanted contaminants, whether it is being present or not. That every day they have to check, and then only it should be discharged into the water body. And then the increased use of excess fertilizers and pesticides also affect the water bodies, because whenever it rains from the soil, it takes up the fertilizers and pesticides from the homes. It directly goes into the river. So that also has to be avoided. So increased usage of fertilizers and pesticides should be avoided. And it is also advisable to use the biodegradable detergents or biodegradable plastics, like that we can take. And here in some places in India, they have got an practice of throwing the dead bodies itself into the river. Say for us Ganges, river Ganges is considered to be very holy. So those who are getting died in that city of Kasi or Varanasi are considered to directly go to the heaven. So they have got that traditional belief. So for that, many people, what they will do, they will go stay in the place Kasi during their old age, around after becoming 60 or 70, they will go stay there itself, and they will take up, yeah. So the last day of the death day will be there. If it is there, it is considered to be holy. So after everything happens, those people, because many dead bodies are coming in per day, so they will just burn it for some time, and then a half burnt body itself is being thrown into the river. So that and all is yeah. So recently our government has taken a tremendous effort to clean the whole Ganges. That is a river Ganges project itself they have taken up. Like that, the government has to come forward to take up projects to clean and to keep the places around the water bodies very clean. And as well as human, they should have a conscience, and they should also see to that we should not throw the litter or other things in the earth, as well as in the, it should be, that is the garbage, and the other things should be properly disposed, so that we can avoid all these things. That's the thing.
Okay, thank you so very much. Very interesting story at the end.
Yeah.
Actually, will be, I guess, will be raise a lot of questions
Yeah.
regarding the what is it, the different point of view between
Yes.
culture, traditional culture, and scientific knowledge, Professor
Yes.
Gayatri.
Yes,
And we
madam,
have
yes.
to find a way how to
Yes.
solve the problem.
Definitely.
Okay,
Yes.
Prof. Gayatri, once again, thank you very much for the
Yeah.
impressive lecture.
Mm.
However, to limited time, are there any final thought before we end these sessions, Prof. Gayatri?
Yeah, I would like to thank all of you, all the organizers, and other people of University of Brevigio for giving me the great opportunity. And I would like to meet you all soon in person. So please give me an opportunity for that also. And please
Yes.
convey my thanks and regards to all the higher authorities, as well as Dr. Oun, for this great opportunity. Once again, thank you one and all present here, all the participants, as well as the upcoming invited speakers, as well as the resource persons, all of you. Thank you from India. Thank you all.
Oh, you are most welcome. We also thank you very much for your present. Okay. I think, yeah, thank you so very much.
Yeah.
And
Can I leave, madam?
yeah, yeah.
For
So
this
we will meet
photo
in the
together.
closing ceremony, I suppose.
Sure, sure, sure.
Yeah. Thank you again,
Thank you, thank you,
Prof.
thank you.
Gayatri.
Thank you, thank you. Bye
Dr.
bye.
Umi, please
Yeah.
photo together.
Oh, Dr. Gayatri, there is a request from Dr. Uun to have a photo session together.
Okay.
All participants.
Okay. Please, all participants, please, if you are allowed to on cam for a while, so that we can the committee will be able to yeah. Okay. Yeah. Thank you so much. Yeah, it has been done with all the participants as well. Thank you, Prof. Gayatri.
Bye bye, madam. Bye
Thank
bye,
you,
all of you.
Uun, for
Dr.
the
Oon,
reminder. Okay,
Terima,
bye.
terima kasih. Terima kasih.
Oh, terima kasih kembali.
Yeah.
Okay, shall we have a break for a few minutes, or we can directly into the second probably, yeah, keynote speaker. Yeah. I guess I have to continue for the second guest speaker. I would like to know whether Prof. Rafida already. Hi, how are you? It's nice to meet you.
Yeah, thank you.
So how are you?
I'm good. Yeah.
Oh, okay, Rafida. It's nice to have you here again. So
Alright.
now for this second plenary session is your time to have to deliver your material. But before that, please let me introduce you by your curriculum vitae. Do you have to deliver? Please open the curriculum vitae. So I will read it. Yeah. Okay. All right. So Professor Dr. Rafida Binti Othman, for the qualifications, the DPM is from University Pertanian Malaysia in 1998 on the topic of veterinary. The PhD, University Pertanian Malaysia, 2013, in the topic immunology and veterinary medicine, and postdoctoral on Ohio State University, Ohio, USA, and 2019 till 2020 on aquaculture. Yeah, current professional membership is Malaysian Veterinary Council, Malaysian Veterinary Medicine Medical Association, Sabah Doctor of Veterinary Medicine Association, Malaysian Association of Veterinary Pathology. Research area in aquaculture, veterinary medicine, research interest in fish health and disease, immunology, fish production, animal production, field of expertise, aquaculture and veterinary. Previous employment, April 2014 till present, senior lecturer at Borneo Marine Research Institute, UMS. And previously, December 2002 till April 2014, research officer at the Department of Veterinary Services, Sabah. There are a lot of conference that has been attending by Prof. Rafida, as well as the publication, more than 17, and innovation as well, more than 8 innovation that has been achieved. Thank you very much for Professor Rafida in coming at our conference. Now, without further ado, I would like to invite you to deliver your lecture today. Please, time is yours, Prof. Sorry. Yeah. Professor, you're still in mute, I suppose. Please unmute Professor Rafida. Oh, I'm sorry, Professor Rafida. You're still in mute. Prof. Rafida, you're still mute. We still cannot hear you. We still can't hear you. Perhaps we can chat her. Hang on. Hello, Prof. Rafida. We still cannot hear you. I guess you have to search for microphone and select speakers. Okay. Yeah.
Okay, boleh dengar?
It's okay.
Okay, all right. So sorry, dia a bit delay. Okay, so thank you and sorry for the technical. I don't know why. I can't hear just now here.
Yeah,
Sekarang boleh ya? Sudah bisa?
sudah. It's
Alright.
clear now. Thank
Alright.
you.
Sorry.
It's
Alright.
okay. It's okay.
Okay, thank you. Thank you for inviting me to join this conference of Aquatic Science International Conference for the second conference, right? So today I will present on biosecurity management in aquaculture, on monitoring health and ensuring sustainability. Next slide.
Next, please. Yeah.
Okay. So my talk today will be content of introduction, understanding what are biosecurity in aquaculture, in a global context, principle, what is the technology and innovation that might be helped into reshaping the biosecurity in aquaculture, the best practice and strategy, the environmental sustainability, the future direction, and the impact of economic and social if we are not following a good practice of biosecurity in aquaculture. Okay, next. So I'm sure that most of us here are dealing. I mean, we are aware of aquaculture. We know that aquaculture is referred to any breeding or rearing or harvesting of animals, plants, and all type of water environment. And aquaculture is one of the food production sector that is growing rapidly. As for we can see here, this is the world aquaculture production for since 1991 to 2020. We can see the rapid increasing growing of this industry, which is it about maybe six time from the starting of aquaculture in this world. Next. So we know that aquaculture is emerging as one of the most viable and promising sector, which we can increase the need for animal protein in providing nutritional and food security to human. We can see here the global aquaculture market as compared projected for 2027 from as compared to now, which is it can contribute to 41 billion US dollar, and in a few years forward, it will increase to 56 billion. So aquaculture is very important sector, which is now we are everyone like are talking about food security, sustainability. So aquaculture, one of the sector, a very promising sector that we need to take care of this sector for our future. So what is biosecurity? Yeah, we as academic, I'm sure that we are not, we cannot far. I mean, leave it just without definition of what is biosecurity. So in aquaculture, this biosecurity is just quite new. It just occurred in first discussion in during World Aquaculture Society special session in 1997, which is at that time they are talking about sustainable shrimp farming on emerging technologies and product for biosecurity and zero discharge, because we are aware at that time that we are having quite impacted of shrimp farming due to a disease and outbreak. So later on in 2003, it started like talking about biosecurity in aquaculture through several workshop, publication, and information provided, and a problem with biosecurity being discussed. So the definition in term of agriculture sector, biosecurity is used in starting in 1980 as defined as the sum of risk management practices in defense against biological threats. While for United Nations Food and Agriculture Organization and World Health Organization, biosecurity is referred to as a strategic and integrated concept that contain the policy, regulatory framework, which include in that policy is the instrument, the activities that will be analyzed and manage risks in food safety, public health, animal and plant life and health, and including also environmental risks. While uh, in animal world, animal organization, OIE and FAO, they are referring to animal production, food safety, biosecurity is include all measures to prevent introduction of pathogen, which is known as bioexclusion, and also at the same time to reduce the spread of pathogen or containment. So in general, if we are talking about bio or biology, we are talking about living things and living animals or living plants, and security, of course, protection. So in general, we can talk that biosecurity is life protection. So it involves in many being used in many industry, in human, in animals, and in plant itself. So in aquaculture, all management practices which is adopted to reduce the potential of introduction and spread disease causing organism, whether it is onto the farm, within the farm, or between farm. This is the the main agenda of biosecurity in aquaculture, which is the exclusion of pathogen from the system, which is otherwise it is free, and also if there are disease, then they have to eradicate the pathogen once there are a problem. So biosecurity is include a holistic concept, which is the main agenda is for sustainable of aquaculture fruit production. So as I've mentioned earlier, that biosecurity in aquaculture is a maturing activity. This is very quite new. If we compare to livestock animals, terrestrial animals, for example, especially poultry, they are far well established with their protocols and the system and the measures. So biosecurity in aquaculture still need a lot of apa ni, information to need to be improved. We need more information on disease transmission and how we can manage a disease outbreak and eradication, and to solve problem regarding to diseases. So actually there are three level of biosecurity, which is national or international level, which involve a governmental responsibility. And then we have a regional level, which is a shared or industry responsibility. While on the site or on the farm, there are a responsibility of each company or private farm responsibility to take care of their territorial of their area to make sure they are uh, m- avoid introduction of disease or control any spread any disease that on their territory. So this is why we need aquaculture. Why aquaculture need biosecurity? Because if we are look back at the previous years in history, those early years, the international trade was by sea mostly for international, which is much slower, and normally the volume of export import is only in a small volume. Therefore, only few maybe active ports involved, which is quarantine at national borders was very easy to maintain because maybe it just involved few ports and then the volume also very small. But as we compare today, you can be in Indonesia morning having your breakfast in the morning, lunch in Malaysia, and then dinner maybe in other country. So the transportation very fast in within very fast within a day you can be in a different country. This is same with the commodities transportation, which is much faster and in a large volume. So this also come with risk, which is a greatly increased risk for spreading of pathogen. This is what we are having during COVID, right? Everything have to be stand still in order to contain disease problem. So similar with animal and plant, due to these changes in the mode of transportation, we have a flight, we have big containers to export import our product. So aquaculture operation rely mostly on trade, on various input such as broodstock, forced larvae, fingerlings, and feed. For example, even Malaysia, sometimes we are importing from Indonesia for our juvenile, for our larvae, or sometimes maybe from Taiwan. So the movement of these commodities is very like borderless. Therefore, the high risk commodities has continued with little or no risk management will end up with a severe disease associated at farm level production loss, which is can big loss once you mistakenly bring in pathogen from this trading uh, activities. So, for example, here it mentioned in the news that the salmon import requirements into China muddy the water. There are like there are there are stringent regulation being imposed because we in the era of containment of disease must be uh, taking carefully consideration. So diseases is very economically affected regardless of aquatic species or any aquaculture ventures, whether there are small industry or big investment. So there are significant risks to success due to this ever present of a threat from disease outbreak. In 2014, World Bank reported that disease causing estimated 6 billion US dollar annually for the aquaculture industry on a global scale. While in it is reported in 2000, year 2000, white spot disease caused estimated 1 billion in value losses due to this disease, which is equal to 200,000 ton. So biosecurity is a fundamental concept to prevent and control the occurrence and spread of disease infection in aquaculture system, because these losses is not just affected the animal itself, but once like a big outbreak occurs, maybe some farm have to close down. It also give will affect the livelihood of the community which is involved in this industry. So there are few goals of biosecurity that need to be focused for aquaculture industry. So the major goal is animal management, pathogen management, and people management. In animal management, we have to optimize health and immunity. For people, we try to prevent and reduce or eliminate for the pathogen. Is the aim is to prevent introduction of disease, or if there are disease being having problem in that industry, we must try to reduce, and the best one is to eliminate, uh, like culling elimination being normally practiced in livestock industry. Once they detected, there are very strict, mass culling being a normal practice in livestock industry. And then at the same time, because this is biosecurity, all living things within that industry must be managed, also including the people itself. So we need to educate, we need to manage the staff who involved with the industry, and also the visitor or the client itself. So this is uh, the main target for aquaculture, for biosecurity management, in protecting for industry and for a more sustainable aquaculture. So the use of biosecurity is to reduce risk of disease introduction and to minimize spread on farm or to a new areas, promote fish health. That's why we nurture the health in fish. And we have to protect the investment, because some big industry, they invest a lot of money in their system and their industry. So this is about investment. So at the same time, we have to protect against new disease. Now is very famous with emerging and re-emerging diseases in aquaculture. And also at the same time, because we are producing food for food security, and there are also we need to protect human health by preventing. We know that in disease, animal disease, there are zoonotic diseases also, that we need to protect human health in terms of disease infection, also in terms of food security. So biosecurity plans is a very various. It depends on the type of production, whether it is a big scale, whether it is intensive or just a small scale. Uh, Depends on a species what you work with, because aquatic animals, organism, aquaculture involve plants, also fishes, also mollusks, shrimp, and so on. So there is no one size fits all solution for biosecurity in aquaculture. It depends on a species, life stages, and the range can be simple and quick. For example, providing food dip, disinfectant, signage in the farm. But there are also need economic investment effort. For example, we need to produce, prepare dedicated quarantine equipment, quarantine area, all those in taking care of these animals. So somehow in planning biosecurity for aquaculture production, it must become with a cost benefit analysis. So for example, this is a big aquaculture farm that I visited in China. They are producing a grouper. So of course they are really put a big investment, and they are also uh, a good place in place of biosecurity measures in this farm, because they cannot afford to lose their investment by by a small mistake of these biosecurity activities not taking care well. So, why we need to develop biosecurity plan? Because we need healthy animals. Because healthy animals means that it will increase performance, increase production, and then at the same time, we need to have a mitigation plan for disease transmission or amplification within or between farm, because we need to take care not just our area, but also other farm and our surrounding. Sorry. So early disease detection is very important for the farm able to reduce impact if there are outbreak or there are disease introduction. And then freedom of disease is very important in in production, and also to market your product, market access. If we know that, for example, sorry, I lost my hand. The WTO, World Trade Organization, or WHO, or European, they have very strict requirement for us to penetrate their market. So we know that aquaculture is mainly produced by Asian country, and to penetrate the market of this Western country, they're very strict. So the freedom status of from a specific disease is very good for marketability, and at the same time, having a biosecurity plan also very good for quality control system with the health management and quality measures, quality management. So also very important for facilitate in translocation. For example, you have to import from one area, from one country to another, with proper biosecurity in place in your farm. It will be much easier to move your product to other area. And part of international trade requirement, they are not, because you know that in this requirement international, you not just make a statement. There are need you to prove that you need to provide with documentation that you are practicing, or you are having measures in place for disease containment and control. And at the same time, biosecurity also should be integrated with broader risk management, which is we are talking about workplace, food safety, environmental management. For example, if you're aware of the ASIC, which is Aquaculture Stewardship Certificate, there are, if before this we are talking about GAP, good aquaculture practice, but this ESC is much higher. They are not just looking into your product, but also they are looking how you deal with the environment, how you deal with your human right, your people that work in the industry. So according this global aquaculture product trend, the product of market for aquaculture is increasing from 39 billion in 2022 to 56. So this global aquaculture product market is projected to a big value in growing. So there is a need for us to strengthen our capacity and capability in our industry by having a good biosecurity practices. So as I mentioned earlier, that aquaculture is one of the sector in food production, which is a very fast growing producing food, which is one of important issue now is about food security, because our population now is projected in 2015 we will have about 10 billion people. So with current practices, if we don't really look into how we can mitigate of diseases, how we can mitigate with all the climate changes and issue, it is very unlikely we can fulfill this uh, uh, the huge number of population projection, uh, because we might be not enough food to supply with our uh, keep increasing population. Therefore we need to take care in our industry how we can protect and how we can make it double or triple to meet the requirement. Okay, all right. Okay, another 5 minutes. So we are feeding the future, feeding the world of this 9.7 billion by 2050 is a very challenging, in a very tough, which is we need to collaborate with farmers, food processor, with dealers, researcher, technical specialists, even our leaders, to come with a very good strategy to meet this demand. Okay, next. This a farm. Just Sana. So the principle in biosecurity is you have to identify the hazard. What is a disease transmission? What are the risk factor to the farm? Where's the location? And then you have to assess the risk, the impact to the farm, and to then you determine the measures that needed to be prioritized.
Next, please.
Next, please. So, in biosecurity, there are three intensity level, which is specific pathogen free, which you are dealing about vaccine in lab, but and then it primary breeding industry, mostly you have to take care of your because you're breeding your aquatic animals, and in commercial grower level. So different stages or different level of production will be having a different biosecurity, but the disease control will be either involve all or any one of these quarantine. You need to be have a traffic control, vehicle equipment control, vaccination and medication. Also testing is very important, and depopulation or eradication. So this is, for example, control of a different level of farm biosecurity risk at the entry level, at the internal, and also at the exit level. So quarantine is extremely important because you need to isolate the pathogen to prevent, if you introduce, prevent introduction or spread of the disease. And sanitation in disinfectant also very important in reduce the number of pathogen and prevent or spread of pathogen. So there are various ways of disease transmitted through water, to wildlife, to equipment, through equipment, even from the stock that you bring in into your farm, for the people also, because we are people also as a vector and feed. So we need to identify which is the high risk, and we need to mitigate what to do. So the risk factor for this introduction and spread in aquaculture may be from fish movement from one farm to another farm, from water source. Maybe the water source that you are dealing with is maybe at the same time next to another farm with very very risky also. And how about your fish health?
Oh. Oh. Prof. Rafida, I think we cannot hear you again. Yeah, you are muted again. Main operator, can you open? Why still mute? Oh yeah. Yeah, we cannot hear your voice again. Rejoin again, sepertinya ya. Ini cuacanya barangkali sana ya, Takian. Hello, Prof. Rafida? No, we still cannot hear you. Belum boleh. Masih belum ter... tadi mungkin kenapa ya? Sudah biasa lancar gitu tahu-tahu putus. Macam mana?
Okay.
Okay, now you're back again.
How long sudah saya tidak ada dalam berapa lama saya tidak?
No, it's okay. You can still have 5 minutes.
All right.
Prof.
Okay. Okay, next. I don't know which slide I'm not, I know. So the concept of this different mechanism which aquaculture activities can affect disease in wildlife and the environment surrounding. So we know that the problem with biosecurity, we need to take care not just the fish itself, but we also communicate with the outside wildlife animal transmission interference. There are interspecies disease being being transmitted. So these we have to deal with in aquaculture. So stock movement, we know that it's very also affect the, we have to maintain the optimum stocking density, because very stressful condition also very not good for health of fish, and at the end it will be more susceptible to disease, and it will be impact the biosecurity itself. So if there are a stress condition, we need to decrease the growth, it will decrease the growth, and so on, and so forth. If we are increasing stocking density, it will be rapid disease outbreak. So we have to play around with this situation. Next. So water source is very important, because we are dealing with animals that in the water. So different from livestock animals, we need to be in contact, but in water, the water itself is a good media for the pathogen. And the water source, the water quality, very very important in aquaculture. So we need to treat water a very good treatment. We need to mitigate the risk factor for disease and pest activities in the farm, because we're taking water from the environment, from the water source, maybe from the river, from the sea. Next. And we know that now at this moment, still most like a small industry, a small scale farmers, they are feeding their fish with trash fish, as compared to pellet fish, which is much more good in nutrient. And trash fish is a risk for disease transmission, because the fish itself uh, maybe as a vector for bacterial loading, especially when they are taking the fish from the fridge and then just put it outside and then just give to the fish. Next. Okay, this just setting a normal setting for cage culture. This is how they keep their feed, which is very very not good, because it might be exposed to wet, humid, and this will spoil the feed. And wildlife, birds and animal movement between farm will bring new disease also, especially nowadays in aquaculture area, there are one farm to another, next to another. This might be another farm, this bird bring fish from another farm, and then stop by to another farm. So in this system, they need to put a net over their pond, or over their cage, or over their tank culture system. So cost benefit, as I mentioned, for example, on the left is a small scale breeding hatchery producing a grouper, and on the right one is a big scale in a hatchery. So investing in aquaculture in for biosecurity to minimize the risk of disease outbreak will be very depending on the location and the scale of the industry. No one size fits all for the one solution. So this is another example. They are lining up their pond, which HDPE currently most prawn farming using this type of farming management. So biosecurity, health, nutrition, genetic, environmental quality must be integrated, managed to achieve a uniform and a low-cost product on demand. So biosecurity is a holistic concept for sustainable aquaculture, which is involve government activities, authorities, public consumer, private industry, producer, even society, and of course us as academic and researcher. So at the same time, there are a lot of advanced technology and innovation, which is, for example, there are optical sensor based on plasmid technology. This will be hopefully will be very good in early detection of any abnormality or any behavior changes in water quality or in the pathogen, any pathogen stress indicator and fish itself. So they are now having this technology to be implemented soon. Okay, next. Uh, in Korea, they are having this smart aquaculture biosecurity digital SIS technology, which is they are targeting for reducing biosecurity shock and risk by improving biosecurity effectiveness in health management for more sustainable practices and enhance environmental protection and also promote food safety. So technology is towards new things that help to improve biosecurity. Yeah, again, like now everyone is talking about AI, artificial intelligence is also now in place with there are new apps in place. There are also robot that will help to clean up the net to check whether the net is any leakage to escape for fish or wild fish to a problem. Next. So various from AI technology, from sensor, even including to a genomic system, biomolecular works, which is this engineer biology. They are trying to produce a good, maybe a good what a fish species, which is potentially can more resistant to disease, more sustainable for production. So they are now working on this molecular or synthetic genomic to producing a better breed. Okay, next. So there are
Are there still
yeah,
a lot, Prof?
yeah. Okay. No, no.
Yeah.
I think it's the few last one. So there are
Oh, okay.
stages in risk assessment, biosecurity. So this is need to be management capacity from bottom up to top down. Need stakeholder engagement for risk management and producer at national approach. So this need both agency and industry stakeholder work together for a sustainable and resilient. Next. So this an example how risk assessment. You need to see what are the risks and then how the risks are low, risk or high. So this need to be assessed in every farm different priority. Next. So in biosecurity also there are need to prioritize environmental responsibility because, for example, in wastewater management regulation, how you treat your effluent and all this contamination, environmental impact will affect the environmental. So now most of certification, they're also looking into this environmental responsibility of every farm. Next. So next. So there are a few challenges. We need to produce healthy seed. We need to prepare for any emergency, for example, climate changes, emerging and re-emerging diseases. We need to have a very good diagnosis, management of disease monitoring, threat of aquatic species, a policy. We need to strengthen our policy and look after at the same time welfare of the animals. Research need to be more strengthened, and research in various area maybe need to find unconventional pathway for pathogen, how they work, and need progressive management. So economic, of course, once you're not taking care of the biosecurity, there will be disease, there will be losses. So losses in mortality, losses in your investment, and losses in treatment due to treatment. So we need to take care, because if there are losses, there are mortality, it will also affect farmers' losses and affect the livelihood of the community itself. Because we know big industry, they are taking many staff working with them. Once the industry collapse, then the whole community will be affected. And at the same time, we need to take care of the environment. So freedom from disease means a maximum profit. All right. Next.
Yeah.
Yeah, we need to understand this is the interrelated correlation between host, pathogen, environment, and disease. Okay, next. So next. So lesson to learn, because for example, in poultry many years ago, they need to produce a kilo, a pound of chicken in 94 days, but currently with a very good biosecurity, they are far advanced in producing chicken, which is they can produce within 38 days with 1.8 FCR. So we need to learn from how poultry industry being developed and growth, because they have a very thorough integration of health, biosecurity, and production system, including nutrition and genetic. So without biosecurity, there will be no revolution in this poultry industry. So again, One Health is one concept that is very trending now, because we're not just looking after the animals, we also looking after the people. And also we need to coordinate between the people and environment, because we get the advantage by rearing this animal, but we need at the same time taking care of this animal, because at the end we are the one that will get the impact from this system. So we need to communicate, coordinate, and collaborating in order to achieve this harmonized health sector in human and animals. Okay, thank you. That's all. Sorry for the my ada
It's
putus-putus. Thank you. That's all.
okay, you're welcome. Thank you very much, Prof. Rafida, for the impressive, comprehensive insights of presentations today. Yeah, due to the limitation of time, so I'm very sorry to tell that probably we don't have that much time for discussion, but I will let probably one or two questions, which is if there are some want to ask or comment or give comment, or maybe just discuss something. Oh, we have one raised hand, Prof. Rafida.
All right.
Okay, to Mr. Fidis Semba Prastia, please introduce yourself. You are from where and deliver your questions. Okay, make
Yeah,
it short, Mr. Fidi.
okay. Thank you very much, Dr. Umi, for the opportunity. And thank you as well to Prof. Rafida for interesting talk. Indeed, regarding this biosecurity is very interesting regarding the sustainable aquaculture in the future. But still, I have a question for you. How far has the application of natural product progressed, for instance, from microalgae or other resources in overcoming outbreaks in Malaysia? And most importantly, in minimizing, the utilization of chemical agents, because we know that it's very dangerous, like antibiotic resistance problem and many more. Yeah, that's it. Thank you for the opportunity.
All right, all right. Thank you for your question. It's very interesting. Yes, in aquaculture, in any food sector, we are trying to reduce the use of any chemicals or even, about antibiotic resistant problem. So currently, we are quite active in a study try to find out alternative treatment by using many products, our natural product. Even my institute, we are currently active with study from microalgae, or from even I myself did study one paper about the effect of microalgae in vibriosis. It does show that it does give good indication to reduce problem. I mean, help in fighting against this vibrio. Uh, Other things like from plant also, we have a lot from plants, even from uh, animal products. We have currently active with this research study, because we try to find a better alternative for our food.
Thank you so much, Prof. Rafida. I hope we can collaborate in the future for this matter. Yes, thank
Yeah.
you.
I have one paper for using this microalgae as anti reducing this vibriosis.
Ah.
I
Look
one
forward to
paper
read
published, yeah.
it. Thank you. Thank you.
Thank you, Prof. Rafida. Okay, maybe one last questions, if there's any, or no? Is it? Maybe I will just deliver one question, short question, Prof. for and the sessions. It seems like biosecurity is very important aspect now in our health. Yeah. So how about the company, industry, or individual that still cannot afford good practices? What should we do? for small scale fisheries or something like that, or individual or home industry? What do you think about it?
Okay, yes, very interesting. Yes, because that's why I told earlier that nothing fits one for all. Likewise, I currently doing a survey among farmers for this KAP, which is knowledge, attitude, and practice for towards biosecurity in their farm. In my survey, I include small farmers to big industry, big players. I found out that most of the farmers, they lack of knowledge, but at the same time they have a good attitude towards biosecurity based on the question, but then they are not practicing pure practicing. So there are gap in this situation. Maybe they don't understand. They need awareness. So I'm going to present this paper in Bali in October.
Oh, wow. Yeah.
Yeah.
Hopefully that you will get
Yeah. If
yeah,
anyone of you join the
feedback on that.
in Bali in 23rd, 24th, to 20
I see.
October. So
Okay.
presenting the knowledge, attitude, and practices among
Okay.
aquaculturists towards biosecurity.
Okay. Okay, thank you very much, Prof. Rafida. Ladies and gentlemen, we have finally come to the end part of this sessions. And before I end up the sessions, Prof. Rafida, are there any final thought or statement before we end the sessions?
Okay, so I know here is mostly maybe aquaculturists. Please take care of our system, our environment, because we are producing food for our future.
Oh, okay. Thank you very much. Yeah. Thank you. Excellent. Okay. So, ladies and gentlemen, we have to really finish the session now. Thank you again, Prof. Rafida, for the amazing presentations. And now I will give it back again to the Master of Ceremony.
All
Thank
right,
you, Prof. Rafida. Have
All right.
a good time and see you
All right.
soon. Okay.
Thank you very much, Dr. Umi Zakia, for leading this session. I hope we all can understand and implement the new knowledge in our future endeavor.
Dear ladies and gentlemen, in this third of plenary session, we will invite Head of Aquatic Resource Management Study Program as the moderator, Dr. Uun Yanuhar. Please, time is yours.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Thank you so much, MC, for time for me. And Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. May I have your attention. The plenary session is about to begin. My name is Uun Yanuhar, and I will be moderating this session. The Excellency invited speaker in Aquatic Science International Conference 2023, Professor Dr. Insinyur Gunanti Mahasri from Fisheries and Marine Science, Airlangga University, Surabaya, Indonesia. I will be read the whole CV. Full name, Professor Dr. Insinyur Gunanti Mahasri, MSc, gender female. The functional position, head lecturer in Airlangga University. Place and date of the birth, Boyolali, September 12, 1960. Office address, Fisheries and Marine Science Faculty, Campus C, Unair, Mulyorejo, Surabaya. And an expertise, fish health, fisheries biotechnology, fish disease, biotechnology, and fisheries with research interest and parasitology. Among her spectacular works are, in 2020, effect of immunostimulant on growth performance of white-leg shrimp, Leptoneus faname, in Pakistan Journal of Nutrition, Scopus Q3. And then in 2020, prevalence, intensity, and histopathology of Ceylani Sobdella arugamensis infestation on grouper rates on different aquaculture system in Scopus Q1. And 2020, immune regulation but not antibacterial activity is a crucial function of hepcidin in resistant agent pathogenic bacteria in Nile tilapia Ocreocomis niloticus in journal Biomolecule Scopus Q1. And finally, in 2020, antioxidant capacity of 5 microalgae species and their effect on head shock protein 70 expression in the brine shrimp Artemia in Scopus Q1. And ladies and gentlemen, standing in front of you all, Professor Dr. Insinyur Gunanti Mahasri would like to deliver her speech under the title presentation, Distribution, Abundance, and Potential of Immunogenic Marine Lice Protein to Increase Sustainable Culture Fisheries Production. I have a few housekeeping notes where Prof. Gunanti Mahasri will be present for 30 minutes. After that presentation, I will open with discussion, have total time for 15 minutes. For participant, if you have any question for the presenter, please raise your hand and say your question. And now, welcoming to speech for presenter, Professor Dr. Insinyur Gunanti Mahasri. Please for speech time for you. Thank you, Prof.
Yes. Thank you very much for the time. Assalamualaikum warahmatullahi wabarakatuh. My honorable all keynote speaker and moderator and all of participants, thank you. Now I will to present my research. It's about the distribution of abundance and potential of immunogenic proteins of marine leeches, Ceylanicobdella arugamensis, to increase sustainable of aquaculture production. As the moderator is, I'm from Department of Aquaculture, Faculty of Fisheries and Marine, Universitas Airlangga. The marine leeches are one of the leeches from the phylum Annelida, subclass Herudinia. There are several genera of marine leeches that can be found in grouper fish and which have been identified based on morphological and mitochondrial cytochrome C oxidase of subunit gain sequence analyzed are Ceylanicobdella arugamensis or Ceylanicobdella arugamensis. The infestation can cause wound at the attachment site, so it can act as a predisposing factor for secondary infection by pathogenic virus and bacteria such as Vibrio alvinolyticus. Pteropdella arugamensis is now known to occur around the Indo-West Pacific region, including Brunei Darussalam, China, India, Indonesia, Iran, Japan, Peninsular Malaysia, Philippines, Singapore, Southeast Africa, and Sri Lanka. In addition, P. arugamensis was also reported to be able to transmit hemogregariness and trypanosome to the face host. The hemogregariness and trypanosomes is two of them is protozoan parasitic disease. The prevalence of the grouper and attacked by leeches by Cheilanicobdella can reach 100% with intensity in 100 individual per piece. Next. The next. Potential of marine leech immunogenic protein as an immunostimulant in fish culture. There are several pathogens have been successfully developed as immunostimulant, including an immunostimulant from the immunogenic membrane protein of Zoothamium penai, and then the second is immunostimulant from the immunogenic protein of Myxobolus coispores, and now I still doing research about the protein immunogenic from Cheilanicobdella arugamensis, and I just successfully characterized of the protein immunogenic Cheilanicobdella with molecular weights at 79.5 until 42.43 kilodalton. So the protein, the immunogenic protein of Cheilanicobdella arugamensis is required as immunostimulant because it has molecular weight higher than 1,000 daltons. Next. The research purpose. Next. The research purpose is analyzing the distribution, abundance, and potential immunogenic protein of marine leeches from several grouper fish in floating net cages to increase the productivity of sustainable aquaculture. Next. Material and methods. In this research, we use grouper seed used in post-larva stage 10 to 12 centimeter, and the stocking density in pond in 15 fish per square meters. And the maintenance of aquaculture for 30 days. Next. Next. The parameter test is R distribution and abundance of marine leads and groupers, phase characterization of immunogenic proteins, Zeylanicobdella parasitic infestation, Vibrio-bacterial infection, and parasitic phagocytosis, phagocytic activity, phagocytic activity, superoxide dismutase, total erythrocyte, total leukocyte, and survival rate in grouper phase Epinephelus species. Next. This research had two stakes. There's two stakes. This one is the to find out distribution and abundance of marine leases, and then the second stage is research on the potential of leach immunogenic protein of Ceylani arucamensis and Pterodella arucamensis for increasing sustainable aquaculture production. The aim of this research is determine the potential good of food species immunostimulant to increase sustainable aquaculture production. Next. 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 The relative abundance calculate based on formula in autumn 1993. Next. Next. Isolation of immunogenic protein marine leads based on Aulani 2004. The first stage is collection of marine leads from grouper phase, and the second, and then sample preparation for isolation of immunogenic protein Zeylanicobdella arucamensis. The third stage is determination of protein concentration by using purate protein assay and treat with UV visible spectrophotometer with a wavelength of 595 nanometer. The fourth is characterization and purification of protein using SDS-based, and the last is production of polyclonal antibody on rapid determination of antibody titer by ELISA method and Western blotting. Next. And then the phagocytic activity test based on Aulani and 2004. The next test of superoxide dismutase enzyme level based on Udiati et al. 2016. Next. Total erythrocyte calculation is Ekawati et al. 2012. And then next. Leukocyte black cell examination and counting blood test preparation based on Zainuddin 2007. Research and discussion. Next. The distribution of marine leaves was found in grouper phase from floating net skates in Lombok, Situbondo, Lamongan, and and Mandangin Madura. The higher in Lamong, the higher abundance is in Lamongan Regency 100%, and intensity of this 204 individual for phase, and the lowest abundance occur in grouper from Lombok at 40%, with an intensity of 32 individuals per space. The most influence the development of Zeylanicoptila arugamensis are salinity and temperature, with the optimum being salinity higher than 32 ppt and temperature of 30 degrees Celsius. Next. This is a morphology of Zeylanicoptila arugamensis with carmine staining. The species have anterior sucker and posterior sucker, and if we invest on the face grouper face, they have the blood for the of the house. Next. This is a morphology of Pteroptila arugamensis under light microscopies from Brunei Darussalam. Next. Zeylanicoptila memiliki hermaphrodites. The next because it's next. Yeah. Now we will see the table of table 1 of water quality infection result from sampling location. In this table, you can see that the temperature, the highest temperature it's found at Lamongan district. There is 13.7 degrees ppt, and the temperature 31. The lower temperature is found at Madura Mandangin and Situbondo deserts, 13 and degrees Celsius. The higher salinity it found at Lamongan degrees 37 degree ppt. Next. Characterization of protein immunogenic Zeylanicoptila arugamensis. You can see the figure that this is result for STS base. We found to high molecular weights by Suwardana, and we found three protein. These are 79.5 and 56.26 and 42 and 40.43. The next. The result of characterizations of immunogenic protein Zeylanicobdela arugamensis. These are the kinds of protein characterized by SDS base and characterization by using Western blot. Both are three kinds protein are immunogenic protein. Next. In the table, we can see that giving dose of 5 and 7 ppm increase to immunoglobulin of grouper until the 21st days of maintenance. They're supporting this Verma and Agarwal, an increasing antibody titer is happened because when exposed to the same antigen, the immune response occur quickly by producing higher level of antibodies. The next. In the table 3, we can see average value of phagocytic activity of grouper in each treatment combination. We use 5 doses and maintenance until 28 days. The higher of phagocytic activity found at given 7 ppm and the days of 21 days is 34.2 in the day of the first 21 days. So the average value is not different with the day before. And there's next. This is a picture of the phagocytic activitas cell phagocytic cell by 1,000 times. Yeah. Next. Increased phagocytic activity of blood is an indicator of increased immune system defined on grouper. The entry of foreign bodies in the host body will be responded to by the occurrence of the phagocytic process. It's supported by Yin et al. 2006 and Shahayilatua 2009. Next. Based on the table 4, based on the table, you can see that the group are given a suspension of the immunostimulant of Zeylanicobdela.
Excuse me, Prof. Gunanti, 10 minutes again. Thank you.
Okay, yeah. 7 ppm was able to last until the 21 days of maintenance with a survival value at 80%. Next. In the table 6, you can see total vibrio count of grouper in each treatment combination that the height is of total Vibrio bacteria, total Vibrio con, on 0.12 to 0.13 CFU lime 10 squares per milli. This moment show that Xylania aeruginoensis, a parasite infestation, can act as a predisposing factor for Vibrio alginolyticus because there is a positive correlation, namely in the higher the number of Xylania of aeruginoensis infestation follow the TPC Vibrio. Next. Next. According to Taslihan et al., the minimum threshold for the presence of Vibrio cone is in water is 10 square per CFU per milliliter. And while the minimum limit for common bacteria in water is 10 square 6 CFU per milliliter. If this threshold is explicit, mass mortality of culture shrimp in the pond can occur. Next, the table 7 data of average value of enzyme superoxide dismutase then grouper treatment. You can see that the higher is found with the given doses 7 ppm and the day 23 days. 23 days is not difference with the 28 days. Next. According at Tupang, when 2019, SODX activity increased in grouper after the challenge test appeared earlier in Hymocyte at 6 hours, and according to Mohan Kumar, 2006, the lower SOD activity may be due to the inactivation of SOD by oxide stress produced by single oxygens. Next. Yep. The table, you can see average specific growth rate of grouper in each treatment combination. The high case of growth of rate in the of grouper, it's found that given the immunostimulant, the dosage 7 ppm and in the 21 days. So if we give immunostimulant from protein immunogenic protein Zeylanicoptila stop in the 21 days. Next. If we look the water quality during culture of grouper, the quality of the grouper rearing water is in normal condition. So next. So in the conclusion of this reason is the high abundance and immunogenic protein content of Cheilanicoptila arugamensis from the four location has a high potential, so that it will be able to support increasing sustainable aquaculture production. The suggestion is further research is needed to overcome pathogenic infection after 90 days of maintenance can be done with a booster and given trout feed and booster at 21 days. Next. This is a publication relation with this research and just mentioned by moderator. Thank you very much. Next. Thank you very much for your attention.
Okay. Thank you for your presentation, Professor Dr. Insinyur Gunanti Mahasri. It's very excellent for presentation today. And now I give for the all participant for inviting question. I have 15 minutes and I give time for the participant for give me to question on or raise hand. It's okay. Thank you for Dr. Umi Zakia. Please time for question for Professor Gunanti Mahasri. Go ahead.
Okay, thank you Bu Un for the opportunity to ask questions. Prof. Gunanti, I wonder whether this kind or the same protein also exists in another animal or invertebrate. Why you choose leech? Is this a specific protein that only exists in leeches, or probably in the same class or something like that? That's for my first questions. And the second is whether I was wondering whether leeches from different places contain the same protein in their skins, or probably from other continent or something like that, or another season country with different season. Is the protein also exists? Thank you so very much, Prof.
Thank you for your question, Dr. Umi. And please, Prof. Gunanti Mahasri, time is yours for the answer. Thank you.
Yes. Thank you, Prof. Zakia, for your questions. The protein immunogenic protein approach in animal vertebrae, the other animal vertebrae, this is there is the same of in the vertebrae in fish, yeah, but there isn't leads in the other vertebrae, animal vertebrae, and leads in this lead maybe is the different of family in when attacks the animal vertebrae, other animal vertebrae. We in this research we use the marine list for Cheilanicoptila arugamensis because since 2015, I always observed about the Cheilanicoptila in many locations, especially in Indonesia. It's we can find all of the time, all of the years, and the prevalence in the grouper is high and reach until 100%, and one fish can in intensity can reach over than five individual Cheilanicoptila per phase. So and the infestation of Cheilanicoptila can decrease of the economic, yeah, and maybe because the clinical signs of the Cheilanicoptila, you can see the lesion of the all of body surface and hemorrhage, and so the lesion is can post and can to be entry post entry secondary infection, maybe by bacteria or virus. So it's damage for the from the face. This is one my reason why I use the marine list Cheilanicoptila. Yeah. Maybe Prof. Zakia.
Yeah. Thank you, Prof. Gunanti, for the answer.
Maybe it's very clear, Prof. Gunanti. Yes. So another question for all participants. Yes, maybe I'm question for Prof. Gunanti. Lintah laut is now known to occur around specifically infection to the grouper fish.
Yes.
In your treatment in the research, abundance of the 400 parasite in grouper that infect for the grouper until occur to the defects on the enterocyte and then the leukocyte and then the increasing the phagocyte effects and then to occur the mortality for the grouper, Prof. Yes, can you explains how to done the infection the parasite to the blood cell on the grouper, Prof? Thank you.
If you can you can see the tabel before I have just mentioned about the activity phagocytosis increase is leukocyte is and phagocytosis is you can see the tabel before. I can send the tabel before about like the increasing leukocyte is a indicator for the increasing the body defense. Yeah, the body defense and if the immunostimulant in giving the immunostimulant with the doses increase the will be increased immunoglobulin for the grouper. And until the 21 days of maintenance. According with Verma, that increasing antibody titer has happened because when exposed to the same of antigen. So in processing phagocytosis, the occur respond immune will more quickly by producing higher level of antibodies.
It's okay, Prof. Thank you for your answer for me. And then another question from Imelda Tidora Sombo from UP student. Hello, Prof. Let me introduce myself. My name is Imelda, a Brawijaya student. Talking about parasite, has Prof ever researched sea urchin? Where I live in NTT, people often consume sea urchin, but after consuming them, the effect is drowsiness. Please, for Prof Gunanti, for explain to Imelda. Thank you.
Okay, thank you for your questions. You give me a question about the sea urchins. Sea urchins, yeah. Sea urchins. But I cannot research about the sea urchins. In NTT, the people often consume sea urchin, but after consuming them, the effect is drowsiness. Yeah. I think the marine leech, and sea urchins is different because the marine leech is not damage for danger for human and organism, the other organism, and the effect of the Cheilanicoptila in human is only happen the decrease of economics, value, economic value, because if we the grouper infested by Cheilanicoptila, the grouper in all of body surface, found the Cheilanicoptila, and the one face is you can found higher than 100% until higher than 500 child individu. So the performance of fish that infest by Cheilanicoptila is decreased. So the consumer is only don't want to buy and to eat them. Yeah.
Okay, enough,
Yes.
Prof Kuyanti. Yes, I think
it's enough. Yeah.
yes, for answer to Imelda, it's enough. Okay.
Yeah.
And then
yes.
I call to Miss Rizky Fadila. Go ahead for question.
Thank you, Bu Uun Yenuhar, for the chance. Assalamualaikum warahmatullahi wabarakatuh, Bu Gunanti Mahasri. I was your student in Erlangga University.
Yes.
I would like ask you two question. I wondering about the specific condition that stimulate the leeches, marine leeches, can infect the grouper. And the second one is, if I'm not mistaken, Prof. Gunanti Mahasri mentioned that there is correlation between Vibrio and marine leeches, right? And I'm wondering, what is the first infection? Fibriosis first, then the parasite infect the grouper, or the parasite infected grouper, then the fibriosis happen. Thank you for the question.
The specific, thank you for your questions. And if I look your face, I know you are alumni from Faculty of Fisheries and Marine Universitas Erlangga, but I'm forget what's your name.
I am Dila, Prof. Gunanti.
Dila. The specific of the clinics of Cheilanicoptila is there are many lesi on all of body surface, and you can found the Cheilanicoptila attached on all of the body surface. Yeah, so if you can see, while since the Cheilanicoptila attacks on body surface, you maybe you will afraid on worry, yeah, because it's not interesting, no, to be not interesting, yeah. And the Cheilanicoptila eat the blood of grouper, so the grouper looks anemia, and we have anemia. Yeah. So if the grouper infested by Cheilanicoptila, the grouper the decrease of decrease to appetite, yes, gitu. So it to be more thin and stress, and to be stress on the face, yeah. So if the stress face maybe is it's easy to infested by Vibrio. The Vibrio, the first infection is by Cheilanicoptila. It's by Cheilanicoptila, and Cheilanicoptila makes lesion, and the blood and the happen hemorrhage, and then maybe hyperemia, and so it and then is post entry the Vibrio. So the first infection is by parasite, and then infection secondary by Vibrio, Alcinolyticus, and etc. Yes. Thank you very much.
Thank you very much for your answer, Prof.
Thank you for your explanation, Prof. Kunanti, for the last question in session now. I would like to say to conclusion the session where the abundant silice that's found in a grouper, both in Lombok, Situbondo, and Lamongan, and Mandangin, where the Cylanisobdella arugamensis or also Pterodella or lintah laut can occur in the water quality, and then can inspection to the infection to face like this the grouper. And then the concentration that height of the superoxide dismutase result indicate the infection in the grouper. And then the potential using a protein from the lintah laut can support the increasing sustainable aquaculture to production. And I would like to Prof. Kunanti can speech in the finalization.
Yes.
Prof. Kunanti.
Okay, yeah. For aquaculture, the disease always present of the aquaculture. So we have to prevent that. Then medicine, the prevention is more better by curations. Medicines by curation. So we always to be careful if we done the aquaculture of fisheries. Thank you.
Okay, thank you, Prof. Kunanti.
Yeah.
And right now, thank you to all our present students, and thank you for attending the session. And ladies and gentlemen, I think enough for this session discussion. That's all her speech. I do hope her speech will be useful. The last I say, terima kasih banyak. Wassalamualaikum warahmatullahi wabarakatuh. Thanks all audience, and thanks for all Prof. Kunanti Mahasri. Terima kasih.
Thank you.
Thank you very much, Dr. Uun Yenuhar, for the insightful discussion. By knowing and understanding the topic that have been shared and presented by the speaker, hopefully it will enrich our knowledge regarding the management of aquatic resources, and also we can expand a future collaboration in terms of education, research, and community service.
Dear audience, before we move on to the next agenda, please kindly have a refreshment, and we will continue our agenda with the next special invited speaker at 1 p.m. Western Indonesian time.
Once again, we will continue our agenda with the next special invited speaker at 1 p.m. Western Indonesia time. Please be on time and see you.
Universitas Brawijaya was established by the President of the Republic.
Ladies and gentlemen, welcome back to the Aquatic Science International Conference 2023. We would like to say thank you very much for your participation.
And now, we will continue our fourth and fifth plenary session. For this session will be led by moderator. Without further ado, please welcome Mr. Haider Jamaluddin, MSI.
Okay, thank you, Master of Ceremony, for the time. Before we get started, I'd like to introduce myself. My name is Haider Jamaluddin, and I will be moderating the third section of ASIC this year. For your information, in this section, we will have two speakers who will deliver interesting topics. For the first speaker, we have Associate Professor Dr. Andi Kurniawan will talk and share his experiences concern about biofilm. As we know, biofilm development inside the culture system found to improve the water quality and also acts as a feed ingredient which is directly improve the fish growth. Before I welcome Associate Professor Dr. Andi Kurniawan, I will introduce him. Mr. Associate Professor Andi Kurniawan, staff teaching of Aquatic Research Department from Fisheries and Marine Science Faculty Universitas Brawijaya. His expertise about microbial aquatic ecology and also biofilm, and also his research interests about aquatic microbiology, aquatic ecology, and water treatment. Thank you. And without further ado, I welcome Associate Professor Dr. Andi Kurniawan. Hello, Dr. Andi Kurniawan.
Hello, hi everyone. Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. How are you doing, Dr. Andi?
Fine, thank you, thank you. Thank you so much for having me in this nice conference.
Okay, thank you. And also, without further ado, Associate Professor Andi Kurniawan will talk about biofilm, and the screen is yours.
Okay, thank you so much, everyone. First of all, it's great honor for me to join in this conference in this special day. Allahumma sholli ala Muhammad wa ala Muhammad, because with bless of this prophet, we can become one of the person, I hope, that get the beneficial of the science. And in this conference, wait a minute, I share my presentation first. Okay, thank you. And in this conference, I will speak about a research that I conducted since 2006, I think. And a little bit provocative. I use biofilm alchemy, unraveling nutrient ion absorption and desorption for aquatic ecosystem equilibrium. Why I choose this word, this special word? Because a microbe is very teeny organism that many people underestimate the function of this microbe in the aquatic ecosystem. Particularly, exactly, I start my study in aquatic ecosystem, in marine ecosystem. My first class that I taught in the Brawijaya University is oceanography. And also me, in that time, the function of microbe in such ecosystem is not so big. However, when I start to take a master degree and then continue to the doctoral degree, take a postdoctoral fellowship in Japan and working for 1 or 2 years as assistant professor in there, I find that microbe, particularly biofilm, is quite interesting. And many of function of this community of microbe may be not elaborate well, because the term of biofilm itself is just found in 1978. It's very young term, and the research about biofilm in the nature maybe just start in 2010. So it's very young focus of research, and that's why I really want to share to you my perspective about the biofilm and what is the one of the most important function of this community of microbe. And this is my outline. I will start with general introduction and then move to the nutrient ion concentration during biofilm forming process, and then ion absorption characteristic of biofilm matrices. Then I will move with explanation about ion desorption characteristic and internal ion environment of biofilm matrices, and then general conclusion. We start with general introduction. Wait a minute. I cannot see the picture in my slide. Is this the same with all you, Mr. Haider? Exactly, I have a picture in here, but why cannot see?
Iya, Pak.
Oh my God.
Either.
Yeah, Pak.
Okay. I'm sorry. Let's we stop first, and then I will try, because if there are no picture in there, it's very difficult to make clear explanation. Why? Oh my God. Wait a minute. I will start. Give me a second. Give me a second. I'm sorry for this uncomfortable.
It's okay.
I just afraid the picture will be not shown. Okay. I get it. Okay. I will share this version. Okay. Let we start again. Okay. This is this one. And I will use my notation first. Okay. This is a general explanation about what is biofilm itself, and I hope this will give some overview for you. We call biofilm as a matrix enclosed microbial population that attach to the surface. So basically, biofilm is community of microbes that form in the surface of various surface. Not only in aquatic ecosystem, but also in our body. For example, in our teeth, should be there biofilm in there. And our studies show that this biofilm is ubiquitous in aquatic ecosystem. They can live in almost all part of aquatic ecosystem and become the predominant habitat of microbe. In our study, we find that more than 65% of microbes in aquatic ecosystem live by form biofilm. That's why if we want to try to understand microbe, but use a planktonic point of view and not use the biofilm point of view, is possibly we just explore about 55% the fact about the microbe in aquatic ecosystem. And this biofilm function various important thing in aquatic ecosystem. They play important role in nutrient cycling. They have ability to purify the pollutant in aquatic ecosystem, and they also become the microbial gene pool. So microbe then live inside the biofilm can exchange the gene. That's why if one microbe have a resistant gene for antibiotic, for example, it's very possible for it to transform this gene to another bacteria then live inside the biofilm. And our study show that the microbial community structure inside the biofilm is totally different with surrounding environment. This fact indicates that biofilm has a specific environment inside. So inside the biofilm matrix, there are a special microenvironment that make the microbial community structure inside different with surrounding environment. This is several kind of biofilm that easily we can find in our daily life. And if we look more detail inside the biofilm, the microbe is just 10% of dry mass. So if we think about microbe live in ecosystem, they live in the matrix of biofilm, the amount of microbe inside the biofilm is not more than 10%. Most of the part of the biofilm property is polymer matrix. And this polymer matrix consists of the biggest part of this matrix is interstitial water. Our study find that interstitial water inside the biofilm is up to 98% of polymer matrix. And this interstitial water, the water that trapped inside the biofilm, has the nutrient-rich microhabitats. This means nutrient ion concentration inside the biofilm matrix are 100 to 1,000 times greater than the surrounding environment. And that's why, in the case of mature biofilm, in this study, we explore the mature biofilm, the biofilm in the mature level. We can find this fact. However, how about the biofilm forming process? What happened with the nutrient ion concentration during the biofilm forming process? And then how the mechanism? What is the mechanism to drive this condition? So we think this is a very important fact that should we understand, and thus we can use the biofilm for many purposes. One of the important objective, of course, in this case, to develop biofilm in the water treatment, for example, or in the sustainable technology for aquatic ecosystem. And to explain that, we create the experiment that focused to explore the nutrient ion concentration during the biofilm forming process. And this is a general overview of the experiment. We put stone and study the biofilm form on that stone, and also study the biofilm form in aquatic plant. In this case, we use reeds, and then we explore the ion concentration inside the biofilm. And then we modified the forming process. We use two forming day sets that different from one and another set around 21 days. In this case, if the biofilm have a universal characteristic, the result in day 0 should be the same, even they different around 21 day. But if biofilm doesn't have the universal characteristic, we cannot find the universal characteristic. We've set this forming process and evaluate in this sampling day, and then compare to the lake water. And what we studied, biofilm wet G, we do microscopy observation of biofilm, we measure ion concentration, we analyze the electric charge, and then we also analyze the total cell count using colony formation ratio. And for lake water, we also do the nutrient ion concentration measurement. And this is the result. This is from the biofilm wet weights, and this is one of the nutrient ion. In this case, we choose ammonium ion concentration. And in the case of a biofilm wet weight, we can see this is the forming process 1, and the triangle is forming process 2, and we can see this result. And from ion concentration, we use 2 y-axis for the biofilm is in millimolar, and for lake water in micromolar. And we get this result. One important result, when we compare to mature biofilm, just need 7 days of biofilm to reach the mature formation. This suggests that formation of biofilm just take about 7 days. And if we compare the ion concentration inside the biofilm and lake water, even it's like synchronized, but biofilm 100 to 1,000 higher than lake water. And how about the increasing after 30 June in here, day formation 42 and 21. This is a very important and very interesting result. And also, before that, also we do the post analyze in another nutrient ion concentration, in this case phosphate, and we get the same result. And for this result, we can understand that, we can understand that the high nutrient ion concentration inside the biofilm is promoted by ion absorption. That's why they are synchronizing pattern between ion concentration inside the biofilm and also in compared to the lake water. When in the lake water high, the ion concentration inside the biofilm will be high also. This suggests that ion accumulated process in biofilm is exactly the accumulation of nutrient ion from the surrounding environment. And then we try to do the microscopic observation and get this result. They are the same trend between day 0 until day 7. And this is the result of observation using SAM. And this is correspondent to this result. However, after that, they are increasing. We analyze in further study and get the result. This is organic and inorganic complexes. So we try to analyze the ecosystem. Why this happened? 3 days before the sample, we get the high rainy season. We get a rainy day. And this rainy day bring the organic and inorganic complexes. And biofilm can also catch this organic. That's why even the forming process in day promising day 2, just in 21 days, it is also used so the same pattern. This means biofilm not only can absorb an organic material like ion, but also complexes organic and inorganic material. And this ability will be important when we try to understand, for example, the ecosystem in the pool, in the stream aquaculture, for example, or another kind of aquaculture. And this is corresponding to this increasing. And then we try to analyze the number of total bacteria and find that the bacterial community inside the biofilm is much higher than the surrounding environment. And there are specific results related to the characteristic of colony formation. In early, in the beginning of the formation step, the formation that we form is very fast, need a less time, and then need a more less incubation time. This result suggests us that the greater number of active bacteria that fast grower exists many in the early stage than the later stage of biofilm formation. And why this can happen? Because nutrient-rich habitats promote the ecosystem, the microhabitat that make this condition possible. And if we think about the nutrient ion have a chart. And we, as a next step, think how this nutrient ion can come to the inside the biofilm. And we hypothesize the process is electrostatic interaction. As you know, the positive electric charge will be connected with the negative electric charge and vice versa. And if this hypothesis really happen, the biofilm should have the electric charge. And then we modify, we make the protocol to measure the electric charge of biofilm. This is exactly the simple method, but very complex to do, honestly. Basically, the principle, we put the biofilm in the electric field. If biofilm have a positive charge or negative charge, it will move to the positive charge. And also, if biofilm have a positive charge, it will move to the negative charge. We measure the movement and then analysis electrophoretic mobility and use that to analyze the electric charge of biofilm matrices. And this is the result. This is the day forming one, and this is the set in forming process two. And we can get all the biofilm in that time have the similar characteristic of EPM, electrostatic charge. One important result, this is the y-axis is so the electric charge. So this is the positive and this is the negative. And from all the biofilm samples, we can get result that show biofilm has both positive and negative charge. And that's why biofilm can attract anion and cation. And if we see there are a great change around pH 4. In the experiment, we measure the electric charge in different pH. So in this case, when we decrease the pH, we put more hydrogen ion, proton ion, H plus to the system. And we can see around pH 4, they are very actively protonation. So from the result, we can see this one. It means they are carboxyl group that very active around pH 4, or in the chemistry term, pKa 4. And this is carboxyl group. So from this situation, we can see this result. Biofilm in the polymer have many carboxyl group, CO minus, and also have the positive charge from the amino group. However, in this case, the amount of negative charge is higher than positive charge. That's why biofilm show the total net negative charge. However, when we decrease the pH, the proton ion H plus attached to the CO minus, and the negative ion become neutral. And that's why around pH 2, we can measure positive charge as a net charge of biofilm. This shows that biofilm has a functional group that bring positive and negative charge, then make biofilm can attract cation and anion in the same time in the water, from the surrounding water. And then we do the PCR DGGE. We do the DGGE and then get this result. And from this pattern, we measure the species. And this is the result. This is the species for the stone biofilm, and this is from the red biofilm, and this for lake water. And from the result, we can see that the community structure inside the biofilm is different with lake water. And if we focus in biofilm, we can see that the community structure inside the stone biofilm is different with the red biofilm. This provides the understanding how microbes form the community structure. And this is the multidimensional scaling if we want to know how the pattern of community structure. From that result, we can get this little summary. Biofilm just need about a week to reach the level of mature biofilm. And then from the very early stage, the nutrient ion inside the biofilm has already high. That promote the community structure inside the biofilm to more dense and different compared to the surrounding water. And this process, due to the ion accumulation inside the biofilm from the surrounding water, that promote by attractive electrostatic force. We move to ion absorption characteristic of the biofilm. We find that there are accumulation of nutrient ion to biofilm. That this make biofilm to become nutrient-rich environment. And now we analyze the detailed mechanism. And to do that, we create the methodology, we create the experiment to explore ion absorption and ion desorption through the understanding of ion absorption kinetics and absorption isotherm, and also analyze the ion desorption process by compare with ion exchange. Because during this research, many researchers in the world said that ion accumulation inside the biofilm happen to ion exchange mechanism. This is the sampling. We use the biofilm on the stone, and then for the physicochemical properties, we do electrophoretic measurement, as I explained before, and we do also acid-basic titration, potentiometric titration, and for kinetic ion absorption, we put the biofilm in solution that having the ion concentration, and then measure the absorption ion and desorption ion during different time. So the time course and amount absorption is important in this set. Another set, we put the biofilm to the solution having the ion concentration, and then increase stepwise, increase gradually the concentration of the ion. And the same, we measure the ion absorption and ion desorption. And this is the result. This is the result from EBM. And this is for the acid-basic titration, potentiometric titration. This is base side, we put NaOH, and this is acid side, we put HCl. And in each drop, we put 0.03 micron liter, so very small amount. So we get almost not just 1,000, but million of the drop. And then we record the result. And this is the result. And this is uptake capacity. So we calculate how much proton ion and hydroxyl ion or OH minus that absorb in specific pH. And we get this amount. They are a peak of the uptake of proton ion around pH 4 that corresponding to carbolic acid. And also they are ion that neutralize OH minus has hits in pH 11, or in this meaning that has a pKb around pH 11, and that is the amino group. So this result proves the existence of carboxyl and amino group. And then how about the kinetic of ion absorption? We use ion exchange as a reference, and then we measure the absorption and desorption in the several time, and get for the ion absorption and also the biofilm happen in very fast process, reach maximum absorption under 1 minute. And this is the result from strongly ionizing resin and weakly ionizing resin. This is common ionizing resin that usually used in the water treatment, and we can get that the amount of absorption and desorption same. Because in the ionizing resin, the mechanism of absorption is, of course, the exchange of the ion. However, what happened to the biofilm? This is for the case of biofilm. In the case of biofilm, the amount of absorption much greater than the amount of desorption. This means we challenge many scientists that said the ion accumulation inside the biofilm is promoted by ionization mechanism. There should be another mechanism. And in our publication, we said that this is the first publication report that said we should evaluate that one. And if we focus in this side to more easy explanation, we can get that they are attractive ion. This is the amount of absorption, and this is the amount of desorption. There are two kinds of ion, attractive ion and also desorbed ion. And the amount of attractive ion is greater than desorbed ion. And biofilm can retain the ion at least in the two parts, in the charged side and the regions between the polymer. And this is the result of absorption isotherm. So the differences is the x-axis is equilibrium concentration, the final concentration when the absorption stop, and the y-axis is the same absorption amount, and we do it for several nutrient ions. And from the result, we analyze with this model. So this is the model of absorption isotherm. And from this model, we can at least to pattern the purple and the blue. If the purple, it means in the absorption process, there are absorption rate and desorption rate. So in this case, absorption rate constant greater than desorption rate constant. This means the tendency of the biofilm to absorb greater than the tendency to the desorb. But if the pattern like the blue one slowly increase, the desorption rate constant greater than the absorption rate constant. And by calculate the ratio between absorption rate constant and desorption rate constant, we can get the value of B. Usually, in the physical chemical term, B is the absorption equilibrium constant. Sorry. And then we use this equation. This is the derivation equation from the Langmuir model. And from this model, by plotting C per N, C is concentration, equilibrium concentration, N is the absorption amount, versus C as the equilibrium concentration, we can get the linear correlation. And if the correlation is correct, it means the absorption follow this pattern, follow the Langmuir pattern. And from the number, we can get slope and intercept, and then calculate how much NM max. NM max is the maximum absorption amount, and also calculate B, the absorption equilibrium constant. And this is the result. This is the absorbent biofilm, strongly acidated resin, weakly acidated resin, and this is the absorbate. We use cation and anion. This is NN max, and this is B. And in this case, magnesium has charge double that ammonium. And also, potassium have a 3 charge. So this means to absorb potassium using 3 charge, whether to absorb nitrate just 1 charge. This means if just charge will function in the absorption, the amount of potassium should be just 1/3 than nitrate. But however, the result for the case of resin, of course, the absorption of ammonium 2 times than magnesium. And this case also for the case of potassium compared to nitrate. But in the case of biofilm, is not. They are different trend. This means the support the finding that ionization mechanism only cannot explain the ion absorption mechanism to the biofilm. And then this is the result. And then if we compare the B value, the value of B in case of biofilm lower than in the case of resin. And also the same pattern we get from the case of anion. This means the ion, when absorbed to biofilm, is not so strong to be attached. It's easy to the microbe to use such kind of ion. That's why after the ion attracted from the surrounding environment, microbe can be easily use that for growth. And this is also the important result that support that nutrient-rich environment inside the biofilm promoted by ion absorption from the surrounding water. So for the summary, we can get this result. Biofilm polymer have a positive and negative charge. And this charge came from the ionization of functional group like a carboxyl or amino group. And this charge can attract ion from the surrounding environment where the ion that has greater electric charge will be absorbed in the more power. And biofilm can absorb not only by ionization mechanism, but also by attractive electrostatic force. And biofilm will put will retain the ion in charge site and in the region between polymer. And to get a clear understanding, we continue the research with ion desorption characteristic. We know that not only absorption, but biofilm can also promote the desorption of ion. And this one important to make the biofilm become the nutrient-rich habitat that supply nutrient from the microbe. And this means biofilm can able to retain and also resorb ion. And to confirm the process, we do desorption investigation. We use biofilm formed on the stone, and the process is simple. We put the biofilm in the distilled water, and then measure the desorption amount. Then we brush the biofilm and measure the ion concentration after several times. And this is the result. This is the desorption amount, and this is the time. And this is before we disperse the metric, and this is after dispersing. And we can get all kind of ion that previously retained in biofilm dissolve more when we disperse the metric. And it's not happen in the case of resin. This also make the differences between biofilm and resin. And we use this logic. This is a little bit physical chemistry and mathematics modeling. This is the condition. I try to explain in the easy way. This is the condition of biofilm. And then we put the biofilm in distilled water. And then there are some ion dissolve the biofilm. And it's not occur in the case of ionization resin. There are no ion detached from the resin. And this is the condition before dispersing, and this is after dispersing. So the ion that not dissolved, still retained inside the biofilm, can be dissolved and make the concentration of ion in the water increase. If we call it M out, we call it M total. And by compare this one, M total minus M out, we get M in. M in is the ion inside the biofilm that not dissolved, just only put the biofilm in distilled water. And then we can calculate remaining ratio. Remaining ratio defined as a ratio between M in and M total. And this is the result. Before that, because electrostatic interaction important, we must understand the characteristic of the ion. So in the case of ion, not just the amount of charge, but also the size of ion is important. That's why we can get the amount of charge density. So charge density is exactly valency divided to the size of the ion. So higher charge density make a greater electrostatic interaction. And this is the result for the condensation ratio. This is the charge density, and this is the remaining ratio. And if we see the result, higher charge density promote more strong interaction. So higher charge density, it means more strongly retained. And not just it, the ion shape, if you can see this term, this is the simple ion and this is the complex ion. Ion shape also affected the ion retaining mechanism. And if we look the result from the condensation ratio, this means the ratio between M in and M out, we get this result. So this is the tendency of ion to be condensated inside the biofilm. And same, this is condensation ratio, and this is the charge density, and we get this result. Higher charge density condense with large extent. And not just higher charge density, but also ion shapes affected the ion condensation. So if we see, this is model biofilm metric. This is M total, and then several amount is M out, and another amount is M in. We can get the ion that goes to the M out is release extent. Then the ion that show more to the M in is retention extent. And then we try to analyze the result. This is the release extent, this is retention extent, and this is the ion. And we can get this result compared to higher charge density. More higher charge density make the biofilm ion strongly retain. And this case, this result is one of the most important of our study that give understanding how biofilm can absorb and dissolve ion, including the mechanism, and then promote nutrient-rich microhabitat that make community structure inside biofilm become the special one. Higher charge density retained by larger electrostatic attractive force inside the biofilm matrix. So, if we conclude the model, this is interstitial water. Interstitial water is mean water inside the biofilm, and this is the model of biofilm matrix. And then we put distilled water, and then we put biofilm in distilled water, and some of the distilled water will come out from the biofilm, and just not only the water, but also will bring the nutrient ion inside the biofilm. Then from this one, we can understand that main factor regulating ion retaining inside the biofilm is electrostatic interaction, where the higher charge density condense with larger extent in the biofilm matrix. Let's move to the general conclusion. First, we can understand that biofilm form just only in 7 days. And this formation, the fast formation, promoted by nutrient ion concentration. And nutrient ion concentration inside the biofilm 100 until 1000 higher than like water. And this condition make the community structure inside the biofilm different with surrounding environment. And this nutrient ion can be high because absorption from the surrounding environment, accumulation from the surrounding environment. And the accumulation is promoted by electrostatic interaction and also the ion exchange mechanism. That can be exist because biofilm have both positive and negative charge. And this condition make the characteristic of ion, particularly related to the charge density, will affect the situation, the retain mechanism of nutrient ion inside the biofilm. So what we can conclude, especially in the water resource management perspective. This is the model of biofilm. Biofilm in the aquatic ecosystem has both positive and negative ion, cationic and anionic ion. And also in the surrounding environment, there are ion that concentration lower than the biofilm. But when the ion concentration in the surrounding water increase, biofilm will absorb. However, when the ion concentration in surrounding water decrease, biofilm will dissolve the ion. And this process happen in dynamical way. From this result, we can get what I said before. Biofilm has important mechanism that promote its changing ion dynamically with outside environment. This means this little organism, when become the community, have a community between them, not solely as individual, but as community, can dynamically stabilize the aquatic ecosystem concerning ion concentration. That's what I want to share to you. I hope give some idea or some some what happened? I really want to say that alchemy, some understanding. This biofilm, this little teeny organism, not also teeny in the function. They have a big function, various function to the aquatic ecosystem management. So when we think about sustainable development, sustainable aquatic ecosystem, the understanding about microbiology, particularly microbe as community, such biofilm is also important. Thank you so much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Dr. Andi, thank you for the sharing the interesting topics. Really interesting for me. That's why I have several questions that I want to confirm to you. For the first, I can say that the mobilization of ion from the outside into the inside, or the inside to the outside, it's so crucial, right?
Pardon me. So
the mobilization of ion from the environment into the inside of biofilm, so crucial, right? And from the inside into the outside. So how if we add some translocase enzyme? As we know, translocase enzyme is a group of enzyme that can increase the mobilization of some molecules or ion through the membrane cell. Have you ever conducted to add some translocase enzyme to increase the significance of a mobilization of ion? Thank you.
Okay, it's very nice question. After we can get understanding, particularly in physical chemistry point of view, we also think from the biologist point of view. In this case, microbe, when form the biofilm, they communicate between them with quantum sensing. And this quantum sensing is very important, particularly in the biomedical point of view, that really want to block the biofilm formation. Let's say that. And of course, quantum sensing is exactly the process of biofilm to produce the macromolecule. It can be protein, it can be enzyme, it can be another molecule, polysaccharide, for example. And we have analyzed the kind of enzyme. However, honestly, we still cannot get the pattern, what enzyme that really working well, that promote the ion exchange mechanism. We just can say that the biofilm produce macromolecule, including enzyme, and make the functional group access the biofilm in the biofilm polymer. And then this functional group promote the ion absorption and desorption. However, need a further step, need next step. Hopefully one of you will continue this study to make the comparison between the kind of enzyme and the process of ion absorption. That's what we read in this time. And I hope many of you can understand because my field of study is a little bit unique. I'm studying physical chemistry to understand biology.
Yeah.
So when I come to biologists, they said, you too physical chemistry. When I come to physical chemistry, they said, you too biologist. And I get master in master engineering. That's why I take engineering for master. However, Doctor of Science for PhD. When I speak to the engineering, they said you to science. But when I said to science, they said you to engineering.
Yeah.
But I really hope my explanation can give you a perspective and get an idea. I can do such research. Thank you. Thank you.
Thank you, Dr. Andi Kurniawan. Yeah, it's really interesting if you're talking about the combination of physical chemistry, organic chemistry, and also biology things. It's so interesting. Anyway, we have a question in the chat box. For the first question from Mr. Devin Mahardika, the question is, biofilms have a many benefits for the aquatic environment. How exactly is biofilm applied in the aquatic environment, especially in the aquaculture industry? Is there any measurable differences? Thank you so much.
Okay, I answer one by one. Yes, we can use biofilm in aquaculture. And not only to purify the water in the water treatment, but understand how microbes, including virus, affect the organism in aquaculture. Because in our study, we get that most of the microbes, more than 95%, live in biofilm. This means, for example, if you use probiotic, if you use probiotic, we should also analyze the microbe inside the biofilm, not only in the water, because microbe in the water is just a little part, less part of the ecosystem dynamic of microbial ecology inside there. And unfortunately, many aquaculturists still give much attention in microbe that live as planktonic microbe. And of course, we can use biofilm in purification treatment, like to absorb organic material or unorganic that not desirable in aquatic system. And the second question, is there any measurable? In the basic principle, the biofilm that live in the natural aquatic ecosystem, like a lake, for example, has the universal characteristic with the biofilm that live in the aquaculture ponds. And we also conducted study to elaborate biofilm in aquaculture environment. Thank you.
Okay, thank you. Regarding to the question, may I ask one thing?
Of course, of course.
Okay, I want to focus more on aquaculture, especially in aquarium. Okay, because as we know, biofilm can consume the oxygen containing in the water of aquarium. Is it okay? I mean, whether it has a side effect on the aquarium environment. Thank you.
Yes, of course, because many bacteria has also also a part of autochthonous organism. So this means they have a chlorophyll, and they also do metabolism as a plant. And when they do metabolism as a plant, in the absence of the solar energy, they will move the photosynthesis process. So this is also very important to understand the whole, let's say, ecosystem process in the aquarium, for example. And many of the planktonic bacteria become a problem when they develop the biofilm. So in this case, to understand the whole mechanism, how the oxygen concentration, dissolved oxygen in the water, not only plankton that should be analyzed, but also microbe that live as a biofilm.
Thank you. And we have Dr. Umizakia already rose hand. Dr. Umizakia.
Yes,
Okay.
Mr. Haggard.
The screen is yours.
Okay, thank you. Thank you so very much. I saw a lot of questions on the chat as well, so I will make it like short, Dr. Andy. This very interesting topic actually, but I will look at its relations with aquatic resources management. First, the formation of bacteria inside biofilms increase along with the time. Is this process infinitive?
Yes.
Will this process will continue to grow along with time? Or so the amount of bacteria will increase as well?
Yes, there. Let's we move again to the physical chemistry. One of the challenge for the biofilm or another attaching organism is water flow. So the power of biofilm to maintain the structure will be affected by the water flow. If there are no water flow, the biofilm will be thicker. But if there are water flow in there, the volume of biofilm, the thickness of biofilm will be also decreased. And biofilm will always develop. And in this case, one of the forming process of biofilm is not only start from attachment of microbe, but for example, I have a biofilm, this is a biofilm, and then there are a water current fast enough to make this detach, and this detach will be attached to the another surface, and then biofilm also forming.
So it will continue then.
Yes, continue.
Because of according to the rule of life, biological processes should be in S shape instead of linear, like a linear line. So it must be something that limited the process, and then they will reach the maximum or saturation for something like that, and then they will decrease. But in this case, bacteria keep continue growing. Will they will grow?
Yes, and of course, as long as the nutrient ion exists, the bacteria will be grow. However, in some condition, the nutrient ion inside the biofilm will be not enough. And in this case, it's not enough. The bacteria will be moved, will be swim, go aside from the biofilm to attach to another surface, and then make the new cycle of biofilm.
I see. Oh, I see. Yeah. Thank you. Maybe this is last questions. How about microbial or bacterial identifications? Has there ever been research regarding this identifications?
We do it presently. My student do it, not me. My student do it. They use metagenomic, and metagenome is very useful to understand and very interesting thing. One of the very important result that I push my student to do that. There are a very close relation between plankton community structure and micro community structure to the Vibrio attack, to the many problem in aquaculture.
I see. Yeah. I was wondering because probably when the bacterial keep it growing, and then probably the identification or the species is also probably more, or just
Yes.
the common one or something like that.
Yes.
So something like that. But
Okay,
okay, thank
thank
you
you.
so very much, Dr. Andi. Thank you, Mas
Okay, thank
Haider.
you, Dr. Umi. And it will be the last question from Mr. Muhari Jadi from BRIN. The question is, how do you apply industry brackish water pond that has a lot of like a sodium, potassium, calcium, and magnesium ions? Please.
Yes, in the brackish water, of course, we also studied the biofilm in marine and also brackish water environment. So the result that I present is result in the lake water. However, we also conducted in the another aquatic ecosystem, including they are still clean or polluted. And how to apply exactly the same, because what they are interesting point. Even the fresh water in natural aquatic ecosystem also have various kind of ion, and this kind of ion in the term of species of ion exactly almost same with the marine or brackish. The difference is the concentration, the ratio between concentration. That's why in the brackish of our marine aquatic ecosystem, the biggest part of ion is sodium, Na+. This is understandable because in such water, the biggest part is of course in ACl. And how to apply? In my point of view, there are no so differences how to treat the biofilm in fresh water and in brackish or marine water in aquaculture, because the principle is same, the condition is same.
Okay, thank you, Dr. Andi Kurniawan. That's the last question because we have a limitation time. Maybe your closing statement about the use of biofilms for the sustainable fisheries and management.
Yes. Okay. Many things that we can develop from biofilm. For example, one of the many of my students also studied how use biofilm as a bioindicator or in biomonitoring. For example, one of the problem in the river monitoring system, ion will move with water, but biofilm can absorb many pollutant in the very short time and retain such pollutant. And then if we want monitor and evaluate factory or another water use utilization, we can use the nutrient ion inside the biofilm as a biomonitoring or bioindicator rule. And also attachment of the microbe in surface is a big problem that make the biofouling, for example, and become a problem for many things, many diseases in aquaculture. And one of important thing, our study have shown that most of the microbe live as a biofilm. That's why it is very possible we cannot understand all of the microbe that we get many problems from microbe in aquaculture because we still not use biofilm concept. Let's collaborate, let's elaborate biofilm so we can promote more sustainable technology for sustainable development. Thank you.
Okay, thank you so much, Associate Professor Dr. Andi Kurniawan, for the really interesting topic for this ASIC program. Thank you so much, and we will continue to the second speaker.
Okay, thank you.
Okay, thank you.
May I leave, Pak Haidar?
Yeah, you may, if you want to keep staying.
I will. Okay. Thank you. I will keep staying. Thank you so much.
Okay, thank you, Dr. Andi Kurniawan. That's it's a nice and interesting topic for us. For the second speaker, we have Dr. Hilary Rigini.
Hi, good morning, everyone. Can you hear me well?
Okay, good afternoon. Sorry, yeah, good afternoon from Indonesia.
Okay, sorry. Here it's
Here
morning.
is already 2 p.m.
Okay.
Okay, how are you, Dr. Hilary?
Fine.
Okay.
So I will share.
Do you mind to turn on the camera?
I didn't understand, sorry.
Do you mind to turn on the camera? Because we cannot see your face.
Oh, so. Sorry, I try it.
You can turn off the background first. It's okay.
You can see me now?
Not yet.
No.
Not yet.
Why? I cannot share my presentation at the moment also.
You can send into the chat box and the committee will help you to share your presentation. I mean your PowerPoint.
I'm not allowed to I think that the previous speaker is still sharing.
Oh yeah, Mr. Andi. Okay. Okay. Just try again to share your screen.
Okay.
Okay. But we are still cannot see your face.
I don't know why. No.
You can choose blur the background in the start video options. You click start video option, and there's some options and choose blur my background. Okay, it's okay. We will continue without your screen.
I'm sorry, but I switch on the video camera.
Yeah. Okay, thank you. I will introduce you by read your short curriculum vitae.
I can start, so?
Wait a moment.
Okay.
Okay, Dr. Hilary Rigini, postdoc at Department of Agricultural and Food Sciences from Università di Bologna, Italy. Her expertise aquatic microbiology and microplastic. Wow, so interesting. And research them management of fungal diseases on horticultural plants, use of aqueous extract and compounds from algae and cyanobacteria against fungal pathogens in horticultural plants. Currently, among the extracted compounds, polysaccharides and phycobilin proteins applied as a treatment to seed studied for their biostimulant activity and their protective role in activating plant defense response. Okay, Dr. Hilary, the screen is yours.
Thank you. So good afternoon, everyone. I'm
afternoon.
I'm a postdoc at the Department of Agricultural and Food Sciences of the University of Bologna. I would like to thank the organizers for inviting me. It is a pleasure for me to be here at this conference. So during my presentation, I will talk about algae and cyanobacteria as sustainable aquatic resources, and I will talk about their potential in horticultural field. So before starting with my presentation, I just want to present my university and my department. My university is the Alma Mater Studiorum, University of Bologna. It is located in Bologna, that is a city and the capital of the Emilia-Romagna region in northern of Italy. The university is divided in campus located in several cities around this region. The department in which I work, as I told you before, is the Department of Agriculture and Food Sciences, and it is the largest one of the Bologna University. So if you like to visit our website, you will also find a section for the events in English language. And here, for your convenience, I have reported the links. My research group is composed by Professor Roberta Roberti, which is my scientific head, and we collaborate with colleagues within the same department and outside. In particular, I would recall the Spanish Bank of Algae, with which we have been collaborating for many years, and which provide us algae and cyanobacteria for our research. Just to contextualize the argument of our research, we work in the field of plant pathology, plant protection, and sustainable agriculture. And our studies have focused on the use of algae and cyanobacteria as alternatives to synthetic products for plant disease management, since these organisms are a source of many bioactive compounds. These are the main points I will discuss during this presentation: an introduction to algae and cyanobacteria, their main uses in several fields, their application in agriculture, and as a last point, a summary of our research on their use for the control of fungal plant pathogens. So, starting with the first point, algae refers to a very diverse and large group of aquatic photosynthetic organisms, which are all eukaryotics. Their classification is very complex, and with the term of algae, we can talk about organisms grouped in Chromista kingdom and plant kingdom. The differences between them lays in the chloroplast membrane typology, and the chromista have chlorophyll C as accessory pigment instead of B as organisms belonging to plant kingdom. Moreover, with algae, with the term of algae, we can refer to unicellular organisms such as the green alga chlorella on the left, up to multicellular algae commonly named seaweed, such as the Clonia maxima, and you see an example on the right, or Sargassum that can reach several meters in length. About seaweeds, they are of great ecological importance within the coastal ecosystem, serving as food, habitats, and nursery for many associated biological organisms. And for example, seaweed, together with the coral reef, mangrove forest, and the seagrass meadows, constitute a coastal offshore ecosystem with complex structures and species diversity. So we can say that their ecological role is very important. In general, algae are classified in several phyla, mostly based on their pigment profile. Among these phyla, Chlorophyta, Rhodophyta, and Oprophyta are the major ones. Within each phylum, algae share the type of pigments that allow them to be distinguished in green, red, and brown algae. There are three major classes of algal photosynthetic pigments: chlorophylls, carotenoids such as fucoxanthin, and phycobilins such as phycoerythrin and phycocyanin. The several forms of chlorophylls and pigments present in algae lead to the difference in absorption characteristics, and as terrestrial plants, seaweeds depend on solar radiation for the energy necessary to carry out photosynthesis to produce carbohydrates and oxygen. The algal distribution in depth in the sea depends on the light, temperature, and nutrients available. In particular, the type of pigment can make possible the adaptation of algal cells to the light ambient conditions. For example, brown algae are typical in the deepest area of the ocean, so they have a low light requirements. And the presence of pigments such as chlorophyll A, C, plus fucoxanthin allow these algae to carry out photosynthesis in this condition. Algae are used in an increasing number of commercial applications. Maybe the most famous is nori alga, that is a dry edible seaweed used in Japanese cuisine, made from species of red algae of the Pyropia genus. Or another example is kombu, made from species of brown algae. Microalgae, for example, are widely used as food and nutritional supplements, with some species being important source of specific components such as antioxidants, pigments, oil, and vitamins. Algae are also a source of compounds used for microbiological medium, for example, agar, that is a polysaccharide extracted from red algae, also used in food for its gelling property. So algae are also producers of secondary metabolites with several properties such as anti-diabetic, antiviral, antifungal, antioxidant, and antibacterial. Then coming to cyanobacteria, they are a group of ancient bacteria that appeared on Earth more than 2 billion years ago. and they were responsible for the rise of oxygen in the atmosphere and oceans, the so-called Great Oxidation Event. Also, this event took place around 2 billion years ago. So they are the responsible of the transition from anoxygenic to oxygenic conditions through the photosynthesis. Throughout their evolution, cyanobacteria became one of the most diverse and widely distributed prokaryotes, occupying many niches within terrestrial, planktonic, and benthic habitats. Their long history evolved in a broad heterogeneity comprising unicellular and multicellular forms, also toxin producers, and for these reasons, they are of special interest to water quality managers because many of them produce taste and other compounds, several types of toxins and blooms. They are widespread in many environments, such as seawater, and in this case, they are the main part of the plankton, the main part, sorry, of phytoplankton, that is the autotrophic part of plankton. So they are foods, a source of foods of other aquatic organisms, such as fish larvae, for example. They are, as I said before, the causal agent of the so-called blooming, in which they become the predominant biomass. And in this case, the water becomes turbid, and we can have several problems, such as water quality, fish death, and if we come into contact with the water, we can have some skin reactions as dermatitis, or at worst even intoxications. We can find them also in extreme environments, such as hypersaline water, deserts, and polar regions. Or another example, in seawater heated from seeping beneath the lava, a situation typical in Iceland, in the north of Europe. You can find frequently cyanobacteria also in fresh water, and for example, you can find frequently them in an aquarium if you have one at home. In which sometimes you can see a greenish film starting to form and covering the substrate and the glass of the aquarium. Some species of cyanobacteria have adapted to live in the uppermost few milliliters of the soil and rock surface, and together with the other microorganisms, they form the soil crust communities. Usually, these microorganisms in these communities are highly tolerant to extreme environmental conditions because they have evolved to withstand extreme environmental conditions such as desiccation, heat, harmful radiation, and, for example, also osmotic stresses. And this crust is formed by the adhesion of soil particles to polysaccharides that are produced by cyanobacteria. And these polysaccharides confer a sort of protection to these communities. There are several orders of cyanobacteria, principally based to the bacterial cell types. For example, Prococales contain coccoidid unicell cyanobacteria and colonial cyanobacteria. The most famous species in this order is Microcystis aeruginosa, that is a freshwater blooming species. Most of the identified cyanobacteria are filamentous species, and They belong to the order of Nostocales, and one of the most famous is Anabena. The highest number of benthic filamentous species are associated with the oscillatorial order that can form complex colony formations. And these communities usually live in or near marine or fresh sedimentary environments down to abyssal depth in the ocean. Many filamentous species are motile, and they are able to glide along their long axis and display photo movement according to the incidence light. Also, cyanobacteria are used in several fields, such as medicine, or also for animal field. And another field is agriculture, and they are source also of natural additives and colorants, namely picobilproteins, used for food and cosmetics. Algae and cyanobacteria could find application also in aquaculture fields. Obviously, it's not my field of research, but it seemed to me interesting to mention it. Here, I reported just a few recent papers that highlighted how these organisms could represent a sustainable alternative to meal and fish oil. And I think that this field needs to be further investigated. And here I reported some pros and cons of use of cyanobacteria rangi in fish feed. First, they are able and at biomass productivity that is higher than that of plants or animals. And they don't need to grow in a fertile land, but they also can be grown in a photobioreactor. Moreover, they contain protein, lipid, carbohydrate, and amino acids. And moreover, they contain readily digestible carbohydrate, and they also are a source of pigments with antioxidant properties and vitamins. On the right, I reported a case study about the Cyanobacterium microcystis aeruginosa used to feed goldfishes for some weeks. In this study, the problem was that this Cyanobacterium is a toxin producer. The toxin is microcystins, and the problem was that this toxin was found in the fish muscle. Moreover, the toxin level for a possible human intake was close to or even exceeded the World Health Organization tolerable daily intake, representing a great health risk for humans. So an important thing, I think they are useful microorganisms, but be careful about the species to use. Besides the production of primary metabolites that are essential for the growth, development, and reproduction, cyanobacteria produce several secondary metabolites that have interesting in biotechnological applications in several fields. These metabolites are related to the adaptation of these microorganisms to the different environments. And in most of the cases, these metabolites are toxins. An interesting aspect of cyanobacteria is that they are able to conduct nitrogen fixation, so to convert nitrogen into multiple chemical forms and to make oxygenic photosynthesis. But however, now nitrogenase the enzymes that is responsible for the reduction of atmospheric nitrogen is extremely sensitive to oxygen. Therefore, oxygenic photosynthesis and nitrogen fixation are not compatible. And cyanobacteria had to evolve a variety of strategies to avoid this problem. So first, some filamentous cyanobacteria can differentiate heterocystis, as you can see in the figure on the left. These cells lack the oxygenic photosystem and possess a glycolipid cell wall that keeps the oxygen concentration sufficiently low for nitrogen fixation to take place. This strategy is known as spatial separation of oxygenic photosynthesis and nitrogen fixation. Other cyanobacteria that don't have heterocystis may temporarily separate these processes by fixing nitrogen during the night. And again, other cyanobacteria used a combination of these strategies, so heterocystis and make the nitrogen fixation during the night, so in that condition. But which is the use of algae and cyanobacteria in agricultural field? I start talking with algae and with the seaweed. And the use of seaweed as soil fertilizer have a long history for their ability to improve soil nutrition and structure. Once upon a time, it was more about about of the coastal areas, but today the use of seaweed is widespread. The benefits of the seaweeds as a source of organic matter and fertilizer nutrients have led to their use as soil conditioners because they improve moisture holding capacity. They promote the growth of beneficial soil microbes. They improve also soil structure, that is very important for the plants, and increase the soil nutrient reserves, particularly the cation exchange capacity, that is a soil property describing the capacity to supply the nutrient cations to the soil solution for irregularly plant uptake. Brown algae, as you can see in this slide, are the most type commonly used in agriculture, and among them, Ascophyllum nodosum is the most researched algae. Beside Ascophyllum nodosum, other brown algae are Eclonia maxima, Sargassum, different Sargassum species, and Laminaria digitata. During the last 20 years, many products based on algae have been developed and commercialized to be used in agriculture. And as you can see in the table, from the same algal species came different products. This because the producers use different approaches for the extractions of biological active compounds. For example, different solvents, different mechanic methods, some enzymes extraction also, so different methods. As I already said before, brown algae are the type most commonly used in agriculture, and this because they are rich in polysaccharides such as alginates and fucoidans, plant growth regulators such as auxin, cytokinins, gibberellins, abscisic acid, and they are a precious source of macro and micronutrient for plants. And all these substances are very useful for plant growth and development. The literature reports also for cyanobacterial beneficial effect in terms of plant growth stimulation and also for plant development. Here I reported, I think, an interesting environment, the paddy field ecosystem that represent a unique aquatic terrestrial habitat, which provides a favorable environment for the growth of cyanobacteria. From the different groups of cyanobacteria, the nitrogen-fixing nostoc genera are naturally found in floating assemblages attached to the plant root surface, and they are able to colonize the root of rice plant. These cyanobacteria, which are of great relevance in the global carbon and nitrogen cycles, are the main contributors to the natural deposition of nitrogen in paddy fields. Their ability is to provide a natural nitrogen source for the plant. And I think also that this point should be more exploited in crop agriculture. On the right, I reported a work showing the beneficial effect on rice growth parameters, such as the length, the increase of length of the plants and yield, expressed as grain per panicle and panicle weight after cyanobacteria inoculation, and without adding further chemically synthesized nitrogen fertilizers. With these figures, I would like to summarize the beneficial effects of seaweeds and cyanobacteria reported on plants in literature, such as seed germination, the establishment and improvement of crop performance and yield, the increase of resistance to abiotic stresses, and temperature, salinity conditions, and also the enhancement of post-harvest shelf life of perishable products. And currently, the research highlighted also the effect of algae and cyanobacteria against plant pathogens, but it is a more recent argument. Probably plant pathology is a little known field for most of you, so I just prepared a couple of slides about it. Plant diseases can be caused by abiotic or abiotic stresses, and among biotics, we can find microorganisms such as fungi, bacteria, mycetes, nematodes, and in this group, also viruses are included in the list. Among these pathogens, fungi are the most numerous agents that can cause disease in plant. Something also about fungi. Fungi are eukaryotic heterotrophic organisms, so they are not able to synthesize all the organic molecules they need, starting from inorganic molecules. So for surviving, they must use organic compounds previously synthesized by other organisms, such as plants. their many structures are hyphae. The branched filaments you can see in the bottom of the slide. And altogether, the hyphae form the mycelium visible inside the petri dishes on the left part of the slide at the bottom. The other structure is the spore, and you can see a couple of images in the middle of the slide. And the spores are necessary for the dissemination and propagation in the environment of the pathogens. And each spore are able to germinate, penetrate the plant tissues, create a new hyphae and new mycelium, and so make the infection process. In this slide, you can see classic symptoms of some of the most famous diseases caused by fungi. For example, on the left, gray mold disease on strawberry, or powdery mildew on cucumber leaf in the middle of the slide, or symptoms of wilt of tomato plant on the right. Until now, the control of fungi in agriculture was mainly based on the use of synthetic products, which caused several problems. The current European regulations require priority should be given to alternative products in place of synthetic ones for plant disease control in accordance with the principle of integrated pest management. This is also relevant at the social level since there is an increasing demand for healthy foods and with low ambient impact by the consumers. At present, only one product based on algae is commercialized in Europe, and this product is Baciplant. It has laminarin as active ingredient, and laminarin is a polysaccharide extracted from the brown alga Laminaria digitata. This product works by increasing plant defense mechanisms against some fungal pathogens. For example, powdery mildew and gray mold on strawberry and powdery mildew on grape wine. Laminarin, the polysaccharide, has been shown to stimulate the induction of genes encoding various proteins with antimicrobial activity. So in this scenario, we started our research on the use of algae and cyanobacteria and their compounds. Here in this slide, I reported algae and cyanobacteria with which we work most. So among the macroalgae, Ecklonia maxima, Ania adherens, and Hydropuntia cornea, as you can see on the left. And among the cyanobacteria, Anabena minutissima and Arthrospira platensis. They are, algae and cyanobacteria are provided by the Banco Español de Algas, the Bank Spanish of Algae, an infrastructure of the University of Las Palmas de Gran Canaria in Spain. And this center is a member of several organizations, and we are collaborating for a BECA project for the promotion of blue biotechnology. In this slide, I reported an example of isolation and cultivation of cyanobacteria, in this case, Anabena minutissima, carried out at the Spanish Center. The cyanobacterium was isolated from a coastal humid basaltic wall, so among a soil crust community in Fuerteventura Island, and cultivated outdoors in photobioreactors. The growth was monitored daily and weekly the biomass was harvested by filtration, washed, and utilized for our experiment. In this slide, I reported just some details of the macroalgae, Eclonia maxima and Aniaderens, with which are working. Aniaderens is a red calcareous macroalga belonging to the Corallinaceae family inside the plant kingdom. The genus Ania is widely present on the Mediterranean and Atlantic coast, and also in the North and Central American coasts. And the typical red color of this alga is due to the phycoerythrin, the major pigment in the red algae. The cell wall of Ania species are particularly rich also in polysaccharides. And for our research, Aniaderens was harvested at Boca Barranco Beach in Las Palmas on the east coast of Gran Canaria. Eclonia maxima is a brown alga belonging to Lesoniaceae family of the Laminariales order, included in the Promista kingdom. The genus Eclonia is widely present in the South Africa coast, and the cell wall of Eclonia are rich of sulfated ramified polysaccharides, such as fucoidans, that represents from 5 to 20% of the alga dry weight. For our research, Eclonia was collected from the coast of South Africa, harvesting only the fronds from the kelp plant and leaving the beds to regenerate the kelp forest. In our research, in our study from algae and cyanobacteria, we usually extract phycobilly proteins and polysaccharides. In particular, phycobilly proteins have already many applications in cosmetics, in foods, and in Europe they were approved as color-safe additives for coloring ingested drugs. And many studies have also shown their antimicrobial, antiviral, and other biological activities at the human level. And on the other hand, we want to investigate their activity on plants. About the polysaccharides, it is well known their role in stimulating the natural defense in plants. And as I said before, laminarin is an example. And as I told you before, is extracted from Laminaria digitata, that is a brown alga, and contained in the plant protection product Baxi plant. For polysaccharide extractions, the powder was suspended in sterile distilled water, the powder of the thallus, the dry thallus, autoclaved for 20 minutes at 100 degrees, and then centrifuged for some minutes. The supernatant was filtered, frozen, and then lyophilized. For phycobilly proteins extraction, the lyophilized biomass was suspended in a buffer solution in the dark. The phycobilly proteins were desalted, filtered, and then lyophilized. And for our experiment, lyophilized polysaccharides and phycobilly proteins were dissolved in water. Here I reported just some details on phycobilly proteins, since it could be a particular topic for you. Phycobilly proteins are a family of colored proteins responsible for the light harvesting of red algae and cyanobacteria. Phycobilly proteins are water-soluble molecules composed of proteins and chromophores called phycobilins that are the light capturing part. They are divided into 4 main types: picoerythrin, phycocyanin, picoerythrochanin, and allophycocyanin, according to their spectral properties. After the extraction, we have characterized the extracted compounds by using spectroscopic technique, in particular FTIR and FT-Raman. And the results obtained with these techniques are spectra, the figures you can see on the left in the slide. And in general, spectra are very specific for each compound. Here, I just reported the characterization of the phycobilly proteins from Annabella minutissima. And with the two techniques, we are able to characterize and identify the functional groups typical of a compounds, in this case, proteins. So both techniques confirmed the presence of the specific functional group of proteins present in the cyanobacterium pigments, and I highlighted them with the arrows, with the colored arrows. In particular, I underlined the shoulder referred to amide primary, secondary, and tertiary presented in the protein chain. Also, these results were confirmed by the spectrophotometer data I reported on the right. I do not go in deeper about the explanation of the two techniques, and for this type of analysis, we collaborate with a group of chemists who work in our department, and they are the specialists of these techniques. But just for your convenience, here I reported some details about these techniques. They are vibrational spectroscopic techniques, so they are sensible to chemical bonds vibration of a specific compound, and they provide a fingerprint spectra, as you have seen for phycobilly proteins. FTIR is an absorption technique, while FT-Raman is an emission technique. So this is the principal difference between the two techniques. These are the pathogens with which we work most. Podosphaera xanthi, agent of the powdery mildew on cucurbits. Botrytis cinerea, agent of gray mold on strawberry and on tomato. And Rhizoctonia solani and Fusarium oxysporum that are soil inhabitants, and they are also included in the so-called soil-borne pathogens. Our research concerns the direct antifungal activity on fungal growth mycelium and spore germination. So if a compound has an inhibitory activity on the development of a fungus. Then we also carry out research on plant or fruit in terms of disease control. So if a treatment with a specific compound lead to a disease symptom reduction, and of which plant mechanisms are involved in the disease control. For example, changes in plant cell wall composition, transcription of genes, and since I talk about plant pathogens, I refer to plant defense genes. And these genes can encode for proteins with several biological activities, such as antifungal. I mean also production of metabolites, both primary and secondary metabolites. And all these physiological changes occur from a complex metabolic responses called pathway in plant, starting with the perception of a stress, like a pathogen, but also the perception of a molecule. For example, a polysaccharide with a treatment, which triggers a cascade of molecular events ending at various levels by all these physiological and metabolic responses in plant. So in our study, we focused on which kind of plant defense mechanisms are activated, stimulated, or increased by application of algae and cyanobacteria compounds against the fungal plant pathogens that lead to a plant more responsive and already prepared to counteract pathogen infection. As I said before, plant response to stresses can include changes in transcription of genes, encoding for proteins with antifungal activity, production of metabolites, and mostly secondary metabolites with biological activity, such as antimicrobial, or metabolites associated with the reinforcement of plant cell wall. This is an important aspect since plant cell wall represents the first line of defense against invading pathogens, and so it represents a physical barrier. If this barrier is broken by, for example, a fungal pathogen, the plant immune system is activated, and the result of the interaction between plant and the pathogen is the induction of a plant response, including the transcription of pathogenesis-related genes, which encode for pathogenesis-related proteins with antifungal activity. And the most well-known and studied are chitinase and glucanase. That are enzymes very important since they are able to degrade fungal cell wall components such as chitin and glucans. One more plant defense response is the production of a variety of important secondary metabolites such as phenols and flavonoids, because they are known for participating in plant protection against stresses due to their antimicrobial and antioxidant properties, and for their role in the reinforcement of plant cell wall. So coming to some results of our research, we have worked a lot on the antifungal activity of phycobiliproteins against Botrytis cinerea, the agent of green mold disease, in this case on tomato fruits. And during this study, we found that the phycobiliproteins from the Cyanobacterium anabenaminotissima significantly reduced Botrytis cinerea colony growth, as you can see in the figure on the left. And also had an inhibitory effect on spore germination, as you can see on the right, evaluated as CFU, colony forming unit. So the treatment of spores with phycobiliproteins were able to reduce spore germination in comparison to spores treated with water.
Dr. Hilary?
Yeah?
Sorry, unfortunately, the time is up.
Okay.
Do you mind to go directly to the conclusion of your presentation? It's okay, it's okay.
So in conclusion, we can say that algae cyanobacteria represent an important source of natural and bioactive compounds useful in several research fields. In my case, from my experience, they have an overall beneficial effect on plants, and their application increase morphological, biochemical, and physiological plant parameters, help the plant to counteract fungal pathogens, and they are able to induce defense responses such as the production of enzymes, compounds with antifungal activity. So sorry for the time, and
it's
thank
okay.
you for your attention.
Okay, thank you for sharing the insightful and interesting topics. Dr. Hilary Regini already told or already shared to us about the utilization of algae and cyanobacteria for sustainable aquatic resources. Okay. Okay, let me go first to ask some question to Dr. Hilary for the first question. Okay, as we know, for nowadays, there are so many potential of algae and cyanobacteria for food and non-food industry development. And I was wondering, I'm asking your perspective, which one is more urgent, for food industry development or for medicine industry development for the utilization of algae?
Sorry, can you repeat the last part of the question?
Okay. Which one is more urgent, for food industry development or for medicine industry development for the utilization of algae or cyanobacteria?
Okay. Yes. Okay. I think that cyanobacteria are the best microorganisms for this kind of things because cyanobacteria, as I said before, are able to produce a large variety of secondary metabolites, including toxins that are of interest in the medical fields, for example, and also for food industry, as I reported, the picobili proteins are already used since they are natural colorants, and here in Europe they are registered as safe additives, for example. So I think that maybe cyanobacteria are the best microorganisms for this kind of development.
Okay, thank you. And regarding to the question, I'd like to go to the next question. When you use the algae or cyanobacteria, you use in a crude extract or in a pure compound from algae for preventing like plant diseases? You use a crude extract or already isolate to become a pure compound?
Well, for picobili proteins, we extract the pure compounds. While for polysaccharides, we used autoclave assisted methods and we extracted them in water. So it's a sort of crude extract, but by which we extract only polysaccharides. And also the two spectroscopic technique, as I showed you before, revealed us that in this crude extract, there are only polysaccharides.
Okay, thank you. Okay, for your information, we have Dr. Umi Zakia already rose hand. Dr. Umi Zakia.
Thank you, Mr.
Yeah, Dr.
Heather.
Umi Zakia. Yeah, thank you so much for the question. The screen is yours.
Okay, Dr. Hilary, thank you so much for your interesting presentations. Do you mind if I ask questions a little bit outside the but still about cyanobacteria, but a little bit outside of the topic that you, is it okay?
Yeah, of course.
Okay. As we acknowledge that cyanobacteria lives in marine environments and usually become dominant in some cases due to pollutions or something like that, or eutrophications. Now, my question is, for cyanobacteria itself, is their life more depending on phosphorus or nitrate, or probably the ratio or NP, the ratio NP. Which one is more limited for cyanobacteria in their environment?
Well,
Dr. Hilary?
I'm not an expert in this sense, but in my opinion, probably I think that the nitrogen could be the substances that can affect in a certain sense their growth. They are able to fix the atmospheric nitrogen, so probably this could be the limiting compounds, in my opinion. I don't know if I answer well.
No, it's okay. It's okay. I was just maybe related my questions to Mr. Heder questions, or later Mrs. Lutfi questions, if there is still time, Mr. Heder, to read it. When we culture cyanobacteria, shall we put more phosphorus or nitrogen?
For this part, it's difficult to me to answer because I'm not, how can I say it? I not follow the growth of the cyanobacteria because it's a collaboration. So the person who follow this part is the Bank Spanish of Algae. And so for the growth, for all the kind of parameters for the growth, they are the specialists. I'm more focused on the application on plant, so I'm a little sorry.
Okay.
Yeah, we get the point that you already conduct a research on the more focus on the utilization of algae and cyanobacteria. Okay, thank you. And
okay, thank you.
Regarding to the condition, may I ask more, Dr. Hilary?
Yeah, if you want, yes.
Yeah.
Thank you, Dr. Hilary. Thank you.
Thank you,
Mr. Heder.
Okay. I ever read some your publication, your articles that you really concerned on the plant pathogen control. Okay, that's nice. I was wondering, when you conduct a research on the plant pathogen control, have you ever conduct by using precision medicine approaching? Precision medicine approaching believes that every single organism in the world has their own up and down regulation to release some genes or some proteins. When you conduct your research to heal some diseases or to prevent some diseases from plant, have you ever used self or precision medicine approaching? Because I believe each plants, one plants and other have their up and down regulations to express some genes.
Okay.
Do you get the point?
I don't know. If you can reformulate the question, I think it's better.
Okay. I mean, when you apply your pure compound from algae or cyanobacteria, you use two for all of your plants, or just in a specific plants?
Well, probably I understand. We are working a lot on seed treatment with these compounds. So we trying to, in some way, how can I say, by the treatment with the seed, we would like to have a seedling and a future plant already prepared for counteract pathogen attack. So starting from the seed, so the first phase during the germination, the establishment of the seedling in the soil, we would like to make the seed and the seedling express some defense responses for against this kind of pathogens. I don't know if I answered well.
Okay, it's okay. I got a little bit insight from your answer. Thank you. Now we are going to the next question from the chat box from Livy, and his or her question about, as we know, in aquatic ecosystem, cyanobacteria included as a harmful algae. But in your presentation, cyanobacteria can make as beneficial product for plant diseases. How the methodology to improve the potential of this algae, especially in industrial scale or to scale up?
Oh, okay.
Thank you.
So about the cyanobacteria that can produce harmful compounds, the bank span of algae conduct experiments that are able to distinguish between cyanobacteria that can produce toxins and cyanobacteria that don't produce toxins. So for our experiment, we used only cyanobacteria that are not producers of toxins, of course. And about the methodology to improve the potential of these microorganisms, we are working at the industrial scale with the bank span of Spanish of algae, because, as I said before, they are the center that are more focused on the growth and to set better the parameter for the growth of these microorganisms in photobioreactor or in a more scale production for a future experiment. So yes, in this sense, we have this collaboration, but we are not the university that are carrying out this part.
Okay, thank you. I think for the question from Mrs. Lutfi, same answer, right? Because you didn't conduct to cultivate your cyanobacteria, you only focus more on the utilization, right? Because she asked you how to cultivate the microalgae, especially Dunaliella, because she already conducted, but several times failed. She asked how to overcome the problem.
Okay. As I already said, this part of the growth of these microorganisms is conducted by the bank span of algae. But if you want, you can read a review we have written with the director of this center, and in which we have also reported some details on the on the growth for growing cyanobacteria, for example, in photobioreactor or in other ambient. So probably in this review you can find some suggestions for that can answer this question.
Okay, thank you, Dr. Hilary Rigini. Yeah.
Thank you.
Yeah, muchas gracias. Now we are in the end of the section, but before I close this section, I want you to give a closing statement about the utilization and the potential of algae and cyanobacteria for sustainable aquatic resources and management, please. Dr. Hilary?
Yeah, sorry.
Please give a closing statement about the utilization of algae and cyanobacteria for sustainable aquatic resources.
Yes, as you have seen in my presentation,
yeah,
you have seen that algae and cyanobacteria are a very precious source of bioactive compounds useful in several fields, from aquaculture to medicine and agricultural fields. And they are able to, from my experience, to have an overall beneficial effect, in my case, on plant by increasing several morphological and biochemical parameters. So they are microorganisms with a great potential in biotechnological application. So we need to go further in the research in all the fields.
Okay, thank you, Dr.
Thank you.
Hilary.
Thank
Okay,
you.
okay. For your information, tomorrow we still have other agenda. If you are available, you may join for the second day for this ASIC program. Once again, thank you. Muchas gracias.
Okay. Thank
Have
you.
a nice weekend.
Thank you.
Thank
you
you.
very
And
much.
okay, I end this section. Thank you for your participation and your attention. Wabillahi taufiq walhidayah. Wassalamualaikum warahmatullahi wabarakatuh. Mr. and Mrs. Master of Ceremony, time is yours.
Thank you very much, Mr. Heder Jamaluddin, for the nice discussion. Hopefully, this will give us more insight and expand our knowledge in the world of aquatic science with the purpose of creating greater ideas and greater service to society.
Excellencies, participants, ladies and gentlemen, we are approaching the last part of our agenda, the closing of day 1. We thank you for all the eager participation that bring life to today's conference.
Alhamdulillah, our agenda is running well. Thank you very much for the speakers, moderator, and all participants who have been a good participant in this event. Hopefully, we will gain important knowledge regarding sustainable management of aquatic resources.
As human beings, we realize that cannot avoid the mistake. We apologize for all the mistake while hosting the event on this first day. And before really end our meeting today, we will announce the agenda for tomorrow.
The agenda will start at 8 a.m. Western Indonesia time.
The first thing on tomorrow's agenda is parallel presentation,
followed by plenary session.
And we will finish at 4 p.m. Western Indonesian time with the closing ceremony. And we will listen to the closing remark by Dean of Fisheries and Marine Science Faculty, Universitas Brawijaya, Indonesia.
We would also like to remind all the presenters in tomorrow's event to please submit their PowerPoint presentation to the shared link in WhatsApp group. Once again, we would also like to remind all the presenters in tomorrow's event to please submit their PowerPoint presentation to the shared link in WhatsApp group. That's all for tomorrow's agenda.
See you tomorrow. Keep healthy and have a good day. Wassalamualaikum warahmatullahi wabarakatuh.